Good morning, everyone. We will start soon, but before starting, just a quick announcement. There will be probably an emergency test in the morning. So if you heard some sounds, just keep calm. And, we can stay here. We don't have to do the emergency test. So go on with our meeting. Thank you.
Good morning. Di Giulia. Good morning. Let me to briefly introduce you the speaker of today, that is Giulia Capotorti. She is a plant scientist and is a professor at the University of La Sapienza in Rome. Giulia is an expert of land size classification from ecoregion to land unit. And she is and has been involved in several projects. Is applied project focused on the determination of restoration strategies, in particular vegetation restoration action. And restoration priorities are also the topics of her today lecture. So thanks, Giulia, and the stage is yours.
Now I have the microphone. Good morning. Can you hear me? Yes. Thank you for the introduction and thank you for the invitation to this stimulating meeting. So today we will address some new vision about an established rationale, that is, that we need ecosystems for our well-being, and also that ecosystems have their own need to be maintained. What is new with respect to this established rationale is that ecosystems have not just to be protected and conserved, but also, as we can see, within the framework of the CBD, Global Biodiversity Framework, and also in the vision of our European biodiversity strategy, is that ecosystems have also to be restored, as you can see in the highlighted pillars of these two international strategies. So let's try to understand why protection and conservation are not enough, why we should go beyond this measure. These are the examples from our country. We have a lot of protected areas and a lot of Natura 2000 sites in the country that together cover almost 21% of the land. So we are quite protected with respect to nature, but these protection measures have some limits. What kind of limits? What we can see here is the National Park of Vesuvio. And a few years ago, we tried to understand if national parks in Italy act well with respect to the maintenance of landscape quality. And we saw that, on the average, the system of national parks effectively conserved landscape quality with an indicator, the landscape conservation, with landscape conservation values, that is always above the national mean values. And this value was maintained in time, so the landscape quality is increasing within the national parks. Nevertheless, some parks, as the Vesuvio one, have lower values. So it is not so sure that the protection measure guarantees a good status of nature inside the parks. And also, if we go more in depth, the patches of ecosystems within the parks are not always well conserved. These are two examples from the Gran Paradiso National Park and again from the Vesuvio National Park. In the parks of the mountain ranges, we have a good conservation status, while in the parks of the terranean coasts, especially, we have an impact from buildings, from road infrastructure, so an impact of land take also within natural ecosystems, within national parks. And these values, in some cases, overcome 2% or 3% of the surface of these patches. Moreover, what does it happen between these parks? Parks can be considered as isolated patches within the overall landscapes, and we need to also improve the quality of the rest of the landscape. And especially, we have to consider the overall quality of the landscape matrix that is mainly made by agricultural surfaces and urban areas. The improvement of quality of the landscape matrix is the only tool we have at disposal to guarantee the dispersal of species and also the movement of species under climate change scenarios. So, go beyond protection is urgently needed, especially in countries like ours, that is under inhuman pressure from many centuries. And by taking an analogy from climate change mitigation initiatives, we should say that we have to go beyond prevention. In the case of climate change, we have to reduce greenhouse gases emission, but also treat. So we have to capture carbon from the atmosphere, better if through nature-based solution. And the same applies to biodiversity at all levels. I mean at the genetic, species, and ecosystem level. We have to prevent, this is the case of the conservation of residual individuals of an abies that is critically endangered in Sicily, and treat, so assist propagation, assist recovery. Always better if based on sound scientific knowledge, and so the paradigm that a good knowledge is essential to effectively conserve is becoming a new paradigm that a good knowledge and also proper investment are essential to effectively restore. And the main global reference initiative in this aspect is the United Nations Decade on Ecosystem Restoration. This initiative is devoted not just to natural and outstanding ecosystem types, but to all kinds of ecosystem types, both terrestrial and aquatic and also marine, and also to the ecosystems that are strictly joined with human activities, so farmlands and urban areas. Again, still, by considering integrated knowledge importance. Yesterday, we talked about indigenous knowledge, but also traditional knowledge have to be combined with our scientific approaches. And also, our science should be devoted also to applied problems. So we have to provide best practices and innovation. The Restoration Decade is conceived to act in restoring ecosystem and our overall type of systems by a restorative continuum. It means that we have to act on pressures, so reduce impacts, but also rehabilitate, remediate, so manage in a proper way our landscape, and also act with proper ecological restoration actions. That means put on the way of recovery the natural and semi-natural system. The ecosystem restoration is the overall ensemble of these different actions. And to support ecosystem restoration, the IUCN promoted the use of red lists of ecosystems. So what are these red lists? They are based on the assessment of both distributional, so extent, and functional symptoms of collapse of ecosystems. Thus, this assessment will be able to inform claimed restoration measures, as we saw before, by determining in a correct way why ecosystems are at risk, where restoration is of primary concern, and what actions should be taken. The overall procedure for compiling a red list of ecosystems is, first of all, to classify, identify, and describe ecosystem types, then collect data about pressures, and try to assign ecosystem risk according to a set of criteria that includes change in distribution in different time ranges, restricted distribution that is quite similar to the topic of endemisms at the species level, and environmental degradation, biotic processes that can cause disruption of functions in ecosystems, and also, at the end, quantitative risk analysis that it is quite a modeling exercise to assess the future risk for each ecosystems. So the criteria and the categories are quite similar to those adopted for species assessment, but with some adjustment, obviously, especially as regards extent. So we have to combine severity classes, relative severity of pressures, with the extent of ecosystem that is affected by those pressures. And also in the terminology of risk, we have, again, as for species, vulnerable, endangered, and critically endangered categories, but then we talk about collapse of ecosystems rather than risk of extinction. So what happened in Italy? Let's go more in depth on this recent project. The Red List of Ecosystem for Italy was commissioned by the Italian Ministry of the Environment to the Botanical Society that involved 14 national and eco-regional experts in this assessment, and also involved the Union of Zoologists for some advice as regards faunistic aspects. This project was thought to complement available red lists that are provided at species and habitat level, and to integrate the knowledge that was already available to implement for the implementation of the EU Biodiversity Strategy to 2020. That is the MAES process. I don't know if some of you have never heard about the MAES process? No? And MAES stands for mapping, so we have to know where ecosystems are, and assessment, so try to give information about condition and capacity to provide ES, ecosystem services. So the MAES already provided a good picture of what is the ecosystem diversity in our country, what are ecosystem conditions, and what are ecosystem services provided. The red list allowed to integrate this state picture with new information about pressures, so the threats, and also on impacts, so the risk of collapse. So starting from the typology, it is the first step of the procedure, we updated the MAES map with a new version, but with the same rationale for the determination of typologies. Ecosystems in Italy were defined according to a reinterpretation of Core-Inland cover categories that are quite general and provide information by structure and physiognomy, essentially. This information was reinterpreted according to environmental and biogeographic information that are overall combined into potential natural vegetation types. So the interpretation of this combined interaction between species into ecosystems and of integrated population with the environment and the geographical sector they occur, provided the ecosystem typology. So as a result, we have 85 types of natural and semi-natural ecosystems in Italy. Most of all are forest ecosystems. Think about the European classification that just foresee 4 types of forests. So we were able to outline the great biodiversity we have in our country. And this is an example of the arrangement of a single type, physiognomic type, that is the beech forest. We determined that we have at least 3 different types of beech forests according to the different bioclimate in Italy. So we have types that occur in the Alps with a continental and temperate climate. We have types that occur in the south, in the mountain of the Mediterranean side, with a Mediterranean climate, so we have different adaptations to these conditions. And we have also a peninsular type that is in a temperate condition, but with a slight decrease of precipitation in summer, and we call this kind of climate Apennine temperate type. So in different contexts, bioclimatic but also biogeographic contexts, these ecosystems for sure act in different ways and are able to provide different kind or different quantity of ecosystem services. And also, as for the assessment areas, we provided a new experimental tool that is not to assess the overall country, the condition and the risk of collapse for the overall country, but for each different ecoregions. What are ecoregions? Are broad geographic areas with similar common environmental features and biogeographic features. And what you see in the picture are the provinces. These are the Alpine, Po Plain, and Apennine provinces within the temperate division, and the Tyrrhenian and Adriatic provinces within the Mediterranean division. These ecoregions have been adopted also for other assessment frameworks, such as the natural capital accounting reports that are provided by the Ministry of the Environment, and also by they became a standard for the territorial official statistics of our National Statistical Institute. So it is a kind to consider not just one Italy, but 5 different Italies. The assessment criteria were retrieved from the general IUCN consideration, but we go more in depth and selected specific threats to be analyzed based on what? On availability over the entire country, or at least over entire ecoregions, based on the availability of spatial explicit data. We need maps for these threats. These threats have to be analyzed a long time, so we have to consider different stages and trends in threats. We must be able to define thresholds. We must be able to determine what is the severity of a threat and what kind of level of severity determine shifts in biotic composition, shifts in functional ability of ecosystems. And then just for few cases, we adopted expert opinion or local knowledge to be upscaled to the overall country. This is an example of how the assessment procedure goes on, and this example is for forest fire threat. So how to assess relative severity? We collected the data for the frequency and incidence of forest fires in Italy that are provided by the official body of foresters with respect to provincial administration units. So we have spatial data, but with a quartz scale definition. Anyway, these data are useful to determine together what is the severity classes for each province. So we have to combine our knowledge about geographic distribution of ecosystem types with this administrative level, and we are able to do such an exercise, so we know exactly for each ecosystem type how many proportion is within different provinces. And so we were able to combine the two assessments, severity and extent, and in this case, the case of Quercus virgiliana forests of southern eastern Italy, this type was considered endangered for forest fires. So this is the overall picture about our assessment. We were able to determine how many ecosystems are at risk in Italy. And this first picture is the first one to determine restoration priorities. Have we a problem with ecosystem? Yes, we have. We have 5 critically endangered types, but no collapsed ecosystem types. The critically endangered are mainly forests, but also psammophilous types, so we have problems with coastal management and coastal conservation. The endangered types are 12 and involve all the macro types of ecosystems, not just forests, but also the aquatic types and grasslands and shrublands. But what is interesting that both these two main levels of risks are of concern for a tiny proportion of our country. So the residual extent of these ecosystems is very limited. So we have also to devote attention to vulnerable and near-threatened ecosystems. Also, these are restoration priorities. Why? Because these types are those that very quickly can shift around the threshold of risk. So vulnerable may be quite simply recovered to near-threatened categories, while near-threatened can easily degrade towards vulnerable types. And these types, as you can see, cover a more significant proportion of our country and a more significant proportion of the residual natural cover. So, under this assessment, we know what type, how many types, but also why. And this is important for restoration because our actions have to address these threats. And declining distribution, so land use change, represent the most threatening factors in our country. It affects 41 types of assessed combination between ecosystem types and ecoregions, and involve all macrotypes, from forest to coast to waters, and in all ecoregions, even though in alpine and Apennine ranges it is less critical. Then we have artificialization. What is the difference? That we consider the fragmentation by built-up areas and road infrastructure within ecosystem patches. And this threat is especially important in the Tyrrhenian, but also in the alpine and poplain ecoregions. Then we have intensive agriculture. We have a huge use of pesticide, a huge use of water, and of mechanical working of tillage of the land. And these set of activities, of intensive agricultural activities, deeply affect ecosystems, especially where in the poplain, obviously, but also in the Tyrrhenian and Adriatic ecoregions that host the wider plain system in our country. And biological invasion, also this is an important threat in the poplain again and the Tyrrhenian ecoregion, and forest fire, especially in the Tyrrhenian side. So we are able to map the occurrence of risk with respect to ecoregions, and here we can see the distribution of the different levels of risk, with the Tyrrhenian provinces that, for example, host 20 types of vulnerable, endangered, or critically endangered types that represent 31% of the region, but especially in the poplain province and in the Adriatic one, we have a wider amount of ecosystem types that are at risk, and more importantly, the residual extent of these types is very limited. So we have lost these ecosystems. More in depth, what are these threatened ecosystems? The critical endangered types include relictic evergreen forests, I mean the Quercus silex forests of the Insubria sector, but especially the residual patches of plain forests of riparian ecosystem in the poplain, and also, as I said before, the psammophilous coastal ecosystems. The endangered types are 22, and they are more different. They include forests, but also grasslands, shrublands, and other types of ecosystems, including the glaciers and the perennial snows that are considered endangered, but with an increasing threat by climate change. And for these categories, we provided some hints about punctual interventions. So we have a limited extension, a restricted geographic space to these systems. And so by considering main threats that are declining distribution, intensive agriculture, and biological invasion, for example, for the Quercus robur plain forest ecosystems, what we have to do, we have to face these threats by reforestation, so we have to improve extent for these types. We have to slow down the impact by agriculture, promote, for example, organic agricultural practices, and also try to put in place active containment of alien species. The benefits will be a range of ecosystem services that could be provided again by these types, and also the conservation of endangered or critically endangered, as in this case, species that are strictly joined with the ecosystem types. Vulnerable ecosystems are instead a wider number. They also include forests, but also grasslands, shrublands, and water-related ecosystem types. And for these types, it is not important to recover extent, so we need not punctual interventions, but broad-scale interventions. What does it mean? As in this case, for the Quercus virgiliana types of Thermophilus peninsular forests, the main threats, recognized threats, include forest fires, but also intensive agriculture and declining distributions locally. So focused actions should involve forest fire prevention, but also assisted recovery after fires. This is a current issue that is under debate on how to assist recovery after fire events. Slow down again impact of agricultural practices, and also, but limited to the ecoregions involved, reforested. The benefits in this case are more landscape benefits, landscape scale benefits, so we can improve the overall quality of our landscape matrix, and so improve functionality and resilience of the ecosystems embedded in this matrix. So, as concluding remarks provided to the ministry, we said that overall the Italian ecosystems are not at high risk, but special attention has to be paid. What? Yeah? Okay. Special attention has to be paid to vulnerable and near-threatened types due to varied threats and to their extent, while for critical and endangered types that have often a narrow extent, we have to put in place a very punctual intervention. And more important, we showed that due to the market heterogeneity of our country, the ecoregional assessments are much more informative than the national one, and that ecosystem types in alpine and Apennine ecoregions are not so threatened as for the Po Plain and the Mediterranean sectors, that critically and endangered ecosystems mainly occur in the Po Plain and in the Adriatic ecoregions due to a set of different combined and very often synergic threats, and that the Tyrrhenian ecoregion hosts the highest number of ecosystem types, which are instead diffusely vulnerable or near-threatened. So we provided sound scientific knowledge on which, where, and why threatened ecosystems occur in Italy. That at the time of the compilation of the red list was a useful information to highlight connections with emerging national recovery and restoration plan measures. These were the measures we identified as strictly connected, but more important is that we provided a general context, an ecosystem context, and also a landscape context to set priorities, to set planning needs, to set targets of restoration that, however, have to be translated into local scale actions. And this was the case, for example, of the NRRP measure of urban and peri-urban forestation, coordinated by the Ministry of the Environment, that involved all the metropolitan cities in Italy, and we provided this context analysis as the first step towards putting priorities for reforestation in our urban systems. But more interestingly now, such an information is useful to inform the National Restoration Plan that is claimed by the Natural Restoration Law at the European level, also because of the connections of these assessments with other policy issues such as climate, agricultural sustainability, and also land take control. Anyway, further knowledge is needed, as always, and so here is our task. The Natural Biodiversity Future Center, as provided in this report. I hope you already know this report from the last year. We have to do a lot for improving our knowledge, for improving effectivity of restoration actions. So, what space for new research? Where should we go with our activity? We can act at the basic knowledge as for typification, mapping, and modeling. We can always improve our knowledge about the state and type of ecosystems. Then we can devote research to threats and devote research, applied research, to responses. So what about typification and mapping? We can improve the classification of types at the national level, and this exercise is already ongoing by an interinstitutional working group that is addressing another European regulation that is about the ecosystem accounting regulation that is quite related to the natural restoration law. Anyway, within this interinstitutional group, there is a lot of space for scientific improvement of our knowledge, especially as we saw yesterday in some of your posters, very interesting. We have to improve geometric detail, but also typological definition of types. Models. We based our classification mainly on vegetation types together with some faunistic information, but we need to better define the characteristic biota of the types, but especially the interactions that occur between biotic components, and also we have to better define processes and flows for the 85 different ecosystem types we have, and build this kind of graphics of synthetic models of ecosystem types. What about threat and risk assessment? We have to improve the spatial knowledge of these threats. We have to improve replication in time for monitoring. And also, and especially, we have to improve our knowledge on critical thresholds. What is the impact of climate change if we don't know how many degrees will affect the composition of ecosystems? What is the impact of alien species if we don't know at which level these species will replace native ecosystems? So we have to improve all the weaknesses in this diagram for making better assessment, and also why not consider additional threats. And as for, again, for threat and risk, we have to improve also field observation. Try always to integrate remote sensing and other kind of inferred knowledge with real-world knowledge. And it is especially true, for example, for the assessment of the criterion that deals with biotic processes and interactions. And also, on the other side, we have to improve the affordability of modeling, especially for estimates and also scenarios. As regards responses, again, we have to move from the general framework provided by this kind of red list to local scale definition of what? Of potential ecosystem distribution in the present day and also for the future. So PNV stands for potential natural vegetation. We have to define models at the local scale. We have to define the reference model, the reference systems, so the reference level of integrity for ecosystem types. Also try to understand how ecosystems change in time by their own dynamics, so what are the expected outcomes of our assisted recovery, and also define landscape context, conditions, and address land use conflicts. And as regards responses, again, we have to improve also restoration practices by testing different models. This is an activity already ongoing within the National Biodiversity Center in Spoke 5 as regards urban recovery, urban ecosystem recovery, but also strengthening monitoring and envisage adaptive management process. We don't know exactly where restoration actions will go, and we have to improve our ability to predict and also to adapt our interventions according to the real outcomes. So have a good research, and I'm available for your questions.
Thank you, Giulia. There's time for questions. There are questions? No?
Quite boring.
So I have a question, Giulia.
Please, Marielle.
The right list of ecosystem for the Italian country, it's gave to us a very important picture of land ecosystem and a useful tool, of course, to manage them. But I'm wondering, do we have any idea about marine ecosystems?
No,
It's a very
it
tough
is a black
question.
spot, yes. The interinstitutional group for the ecosystem accounting also involved an expert team for the marine side. So we are at the starting point of this procedure, and we are starting to typify and map marine ecosystem types. Then we can think about assessment. But first of all, we have to know where these ecosystems are and
So
what are the threats.
at least you are becoming to typify the ecosystem.
Yes.
This is
Typify,
very important.
yes, they are quite, the typification is quite well established by the
No,
units.
but the distribution of
habitat
types.
But the distribution is unknown.
Questions? I'm trusting you, please.
Chiara.
Chiara is our
Chiara is.
is a milestone, you know, of the PhD
Yes.
meeting.
I have a question about the mapping, and I would like to know, like, in your personal experience, I guess through time and through like the, let's say, scientific history of ecosystem typification and also description, there have been many initiatives and many work groups and etc. So in your presentation you said one starting point was the Greenland cover, and I would like to know from you, in your experience, how hard was to start from the Greenland cover and try to collect all the information about the where those ecosystems were and what were the sources, who described that. So I would like to know how was this experience of like collecting altogether.
Yes, it took 3 months of work, and but it was an artisanal exercise. Now, with the collaboration with ISPRA, we are trying to go towards a more automatic procedure to map ecosystems, but again, taking for granted the strength of considering the relationships between actual occurrence and potential vegetation types. So we will be able soon to provide a new map that is based on more accurate geometric definition of coverage with Copernicus, substantially basic data, but still with a ecosystem view, not of just land cover types, but of ecosystem types, so of interacting species and interactive species with the environmental features. 3 months at least. 40 people for 3 months.
Other question? Other question?
So thank you. I go. Ah, no, please. Oh.
There is another question, Julia.
Thank you for the talk. And regarding monitoring activities on the field, like transects or seasonal samplings, which kind of data are best for your the design of ecological ecosystem risk category? So how define you need the data, or you just need the overall view of the species that inhabit and how they change during the seasons, for example?
We can use field data to validate national models, first of all, but then we should use field data, to provide site-level information about restoration actions. So I think the field will come in the second part of the exercise of our task, because we have exactly to know where and how to act in the real world, and we have to do it by knowing what is occurring with what is happening in the field. Anyway, the building of potential natural vegetation map was made available by fieldworks from a huge set of researchers in the overall country. So the basic knowledge is always essential.
Other question? So Julia, I have a last one.
Yeah.
In the National Biodiversity Future Center, several research are going on monitoring and vegetational monitoring too. So I'm wondering how the National Biodiversity Project could or can contribute to the general picture of the status of Italian ecosystem. Do you have any idea? Do you have a return of this research?
I think that we should, in the near future, after the ending of the project, to widen our view. I mean, many of these types are not urban or agricultural. The terrestrial spoke and the marine spoke should, in the future, comprehend the overall country in their monitoring activities. The transects provided by Spoke 4, for example, are just spots of our diversity and of the critical aspects that affect our diversity. So we have to think about the overall country, to think about the overall ecoregions, and try to push the results to improve assessments at the national level.
Thank you. So, no other question? No one? So, we are ready for the coffee break, and thank you again,
Have a nice
Julia.
day.
Sì, per la tavola rotonda ho visto un'email.
Oh, fantastic. We're almost perfect in time. So welcome back from the coffee break. My name is Francesco Frati. I am from the University of Siena, and I am one of the scientific coordinators of SPOC 3 of the National Biodiversity Future Center. SPOC 3, as you know, is dedicated to the study of terrestrial and freshwater biodiversity. A couple of brief issues. First of all, my apologies for wearing my sunglasses. I have a problem at one of my eyes, so I have to keep it protected. I don't want to establish a distance, but it's a real problem at one of my eyes. And the second consideration is how fortunate you are, how lucky you are of being involved in this fantastic PhD program, and especially of having the opportunity to celebrate the PhD day here at the University of Padua. This is a fantastic place. For those of you who were not here in the two previous editions, those, again, were held in a fantastic place at the Botanical Garden of the University of Padua. I hope you have a chance of visiting that Botanical Garden too, which is really great. And but here, this really a dream for all of us who are working in universities, working in research, this fantastic auditorium, as well as the Galilean tribune outside the door. It's really fantastic. So thank you. I would like to thank Mariella Rasotto, who is not here, and all the Andrea, Chiara, Fabio, and all the colleagues from the University of Padua for every year deciding to hold the PhD days here in their fantastic university. So let's move directly to the talks. This session is entitled "The Biodiversity Monitoring and Innovative Technologies." And the first speaker is Chiara Bottaro from the Milan Polytechnic University. Chiara, the rules are known: 12 minutes, leaving 3 minutes for questions and discussion. I think you have to stay here. Okay.
Good morning, everyone. I can stay further away. I'm going to present you part of my PhD research, which is titled Multiscale Mapping of Urban Green Biodiversity. So urban areas are increasingly acknowledged as ecosystems, and biodiversity is key for ecosystem functioning. Healthy ecosystems provide a multitude of services contributing to the well-being of populations. In this direction, efforts are needed in order to protect biodiversity within the urban settings. So efforts can be described as mapping and quantifying to answer the question what is there and how much is there, or also doing a quality assessment, trying to reply how good is what is there. And finally also to be able to answer to the question how is what is there changing. So throughout this presentation I'm going to focus especially on the mapping part and on the possible monitoring frameworks. So the mapping and monitoring can be effectively carried out throughout remote sensing techniques, which is a broad umbrella of different combinations of sensors and platforms that can provide spatial data across different scales and also with different temporal resolutions. However, every combination of platform and sensor have their own intrinsic limitation in terms of spatial, temporal, and spectral resolution. Doing this mapping and monitoring of the green biodiversity, which is mainly the trees in the cities, because with remote sensing techniques we can mainly detect big features, so not for sure arthropoda or insects, but trees. And the trees in the urban settings are very difficult to detect because the state-of-the-art free image spatial resolution is the 10 meters pixel of Sentinel-2 images, which makes it impossible to detect individual trees. This limitation can be overcome by using high-resolution images, which are often provided by commercial satellites or aerial platforms, but they are very expensive. And moreover, the use of UAVs or drones in the cities are restricted because of privacy and security issues. Finally, bidimensional images lack of dimensional information about the trees. So, for example, the height of the trees and the crown size of the canopies, which are often needed for simulations, modeling, for ecosystem services assessment, as, for example, the carbon storage or the air pollution uptake. So the objective of this part of the research is to apply image analysis techniques on high-resolution satellite images to classify individual trees, species, and also use a tree-stereoscopic satellite photogrammetry to derive 3D over the city of Milano. So the datasets for these two parts of the research are as described in this slide. So for the classification workflow, I have the geodatabase of the city of Milan, which provides occurrences of public trees. And to the other side, for the satellite image side, I have a world view 2 image, which is composed of 8 multispectral bands with 2-meter pixel resolution and a panchromatic band of 0.5 meter resolution. For the 3-stereo satellite photogrammetry workflow, I have triplets of images coming from Beijing 3, which is another commercial satellite. And basically, the reconstruction of the 3-dimensional features of the city is possible by acquiring the same scene by different incidence angle from the same satellite. And this allows a reconstruction, indeed, of the 3-dimensional features of the city. And I will validate this data by using a digital terrain model coming from a LiDAR survey from 2010. So this is just to show you the spectral differences between the sensors that I'm using, the 2 different satellites that I'm using, which are WorldView 2 and Beijing 3. So the bands are different, and also the range of the wavelength each band cover is also different. So this is the classification workflow, and it consists of a pre-processing part of the images and then some techniques to extract just the vegetation from the satellite images and to go on with the tree canopy segmentation. Then there is also the part of the classification itself to finally classify the species of the trees in the city, but I'm not going to present this part as I just have some results for the segmentation part. So This is the pre-processing part, which consists in going from the raw digital number, which is often an integer number, to go with specific formula to the sensor radiance, and finally to the top of atmosphere reflectance, and finally after an atmospheric correction, which is in this case was a very simplified approach, the dark object subtraction algorithm, it is possible to obtain finally the physical surface reflectance of the surface itself. Afterwards, I performed a pan-sharpening, which is a technique that allows to resample the resolution of the multispectral bands, which was in this case 2 meters, to the resolution of 0.5 meters, which is the resolution of the panchromatic band. And this allows to enhance the spatial resolution of the images while keeping the spectral information contained in the multispectral bands. This is just the vegetation extraction part, which is very simple. It consists of computing the normalized different vegetation index and then just thresholding the values I know belong to the vegetation and provide a binary mask with no vegetation and vegetation. This mask allowed to perform a segmentation with some mathematical techniques, for example, the mean shift or the inverse watershed, the canny edge. These algorithms are very powerful, and for example, they are able to distinguish between the tree itself and the shadow of the tree, but they do not distinguish individual trees. So as you can see in the image, they just can provide polygons about the patches of the trees, but I do not really have information about single trees. So to enhance this, I would like to test a more sophisticated model, in specific convolutional neural network, which is with the UNet structure, to obtain an individual tree crown delineation. For the tree stereo satellite photogrammetry part, the main output of this workflow is basically obtaining a digital surface model, so including the altitude of the topographic surface, plus the height of the objects, for example, buildings, trees, that exist on the city surface. And once I have a digital surface model, which is the output of this kind of workflow, I can obtain a canopy height model, which is basically the height of the trees itself. So from this kind of product, it is possible by also simple like region growing and local maxima algorithms to extract the height and the crown of the trees in the city that can be associated then to the classification output. And also, since I am validating the data against the 2010 LiDAR data, I would like to assess how the tree vegetation changed in more than 10 years. So these are the triplets I have over the city of Milan, consist in 4 different blocks, and they were acquired during summer 2024. And this is a first reconstruction attempt of the digital elevation model over the city of Milan. And as you can see from the pictures, there is a very well reconstructed part in which is possible to see the features of the city, while there are some other parts that have been not properly reconstructed, and they are very blurry and they do not provide any interesting data. And also, one very weird thing is that the relative height of the objects in the city are good, are reliable, but then it looks like Milan is 1,000 meter high, like on a altitudinal level, which is not the case because it's 200 meters above the sea level and not for sure 1,000. So I have several issues that I need to go through to indeed go for my next steps, which are for the classification workflow, test a more sophisticated segmentation model for the individual tree crown detection, and also finally to classify the segmented tree canopies, and eventually also evaluate radiometric and spectral differences among the sensors. For example, I would like to compare images from the WorldView 2 sensor and the Sentinel-2 sensors, to also assess the model scalability across different sensors. And for the 3 stereo satellite photogrammetry part, I need for sure to solve the digital elevation model reconstruction issues that I currently have and extract the tree height and the crown size of each single trees in the city, and then finally assess how tree vegetation changed from 2010. This is all from my part. Thank you for your attention.
Thank you very much, Chiara, for this excellent presentation. Also, thank you for staying perfectly in time. So we have time for at least a couple of questions. Raise your hands. There's one question there.
Hi. Do you think that both of your part that you presented can be applied also on the field? I'm thinking of the Venice Lagoon, where we have salt marshes that are very close to the sea level, but there are parts that are full of vegetation, other with sand. Is it possible to distinguish these and also maybe have some indication on the height of the salt marshes, because they frequently change due to the tide, but also due to erosion. So it can also become a method to monitor the erosion of some parts. I don't know if it is what you presented can be used in those fields.
Yes, I think for sure it is applicable. Then for what is my experience, I guess all the classification model, for example, and also the segmentation algorithms, especially the mathematical ones, need some kind of fine-tuning based on how your scene looks. Like, for example, if, of course, you have, I don't know, in the urban setting is very specific because you have like patches of vegetation or single trees. So a segmentation approach is feasible, but when you have, for example, like extensive vegetation cover, then it gets really difficult to discern like individual trees, if this is the case. But if you just want to distinguish sand from vegetation and to monitor, if you want to distinguish sand from vegetation, I think it's completely feasible. And concerning that monitoring of erosion, probably the satellite, the 3 stereoscopic satellite approach I have shown is suitable for like the city scale. So if you have like smaller scale areas, I would suggest going for a LiDAR, for example, which is a more detailed three-dimensional data.
Thank you. Time for one more question. Chiara, can you tell us something about the tree recognition part of your work?
Yes, it is very challenging because thinking of arriving to a species level classification is very hard, since there are, for example, I don't know, some differences between like species that are barely detectable by the human eye and also by botanical expertise. So you can tell the differences between some species just looking very morphological differences. And for sure, by satellite, this is not possible to achieve. Like, of course, the vegetation has some like spectral signature, but the multispectral resolution is not yet enough, probably to attain this level of separation of the species to classify botanical species. And while probably the hyperspectral data are more promising in this sense because they provide a deeper spectral resolution of what can be the spectral signature of each vegetation species, and then probably allow to better achieve the species level classification of the trees.
Thank you very much. Okay, thank you again, Chiara. Well deserved. And we move to the second speaker, Eleonora Cresta from the University of Tuscia, investigating ambrosia beetle species and associated fungal communities. Eleonora.
Thank you. Good morning, I'm Eleonora Cresta, a PhD student of Tuscia University, and my project research focused on exotic insect in natural protected area. And today I'll present you the study we carried out last year in the Abruzzo e Lazio e Molise National Park, where we focused on the study of ambrosia beetles in different management beech forests. So the main characters of these studies are ambrosia beetles and bark beetles. These are two groups that we can explain as small-sized beetles typically associated with the forest environments. And the main difference between these two is that bark beetles are xylophagous insects. They develop under the bark and digging galleries feeding directly on the wood. On the other hand, ambrosia beetles are characterized as xylomycetophagous insect. In fact, they don't need directly on the wood, but they feed the symbiotic fungi they bring into the galleries where the fungi can grow. And in general, these species can cause several damage to the host plants in both natural and productive environments, such as the structural weakening or the desiccation of part of plant. But in particular, the entry hole that they use to enter in the tree act as a gateway for the pathogens. And so this can promote the fungal and bacterial infection. So in general, these species don't pose a particular threat to the forest or crops because they actually usually attack dead plants or decayed plants. But some species, due to their ecology, can affect even healthy plants, causing them to weaken and, in certain cases, die. So particularly concerning are some ambrosia beetle species, and in particular the species that we are considered invasive for our country, and they are introduced into Europe due to anthropogenic action. In fact, the ambrosia beetle groups include some of the most invasive species, exotic species in Italy, posing serious threat to the native biodiversity and the functionality of the forest ecosystem, as their behavior can also facilitate the spread of plant diseases. For these reasons, the aim of our study are to determine the species composition of ambrosia and bark beetles in ecosystem dominated by Fagus sylvatica and characterized by different degree of naturality. Also to assess the performance of different management areas within the Abruzzo, Lazio and Molise National Park in controlling invasive ambrosia beetles. And lastly, investigate the fungal community associated with the captured ambrosia beetle species. About the monitoring activities, this was carried out in the Abruzzo National Park, where in particular we identified 8 different sites, 4 of all-ground beech forest and 4 of management beech forest. In each site we placed 3 cross-vein traps, here in the picture, directly on the plants. And these traps were made attractive using ethanol at high concentration, 95%. The traps remained filled from May to October and were inspected once a month for the sample collection and the ethanol lure replacement. So, in each sample, we collected and selected the ambrosia and the bark beetles, and each specimens were identified at 2 species level using a stereomicroscope and the dichotomy skis. In other, we did another analysis in order to characterize the isolation of the fungal community. So, some of the ambrosia beetles specimen trapped in July and August was used for this analysis, and the insects were before sterilized using a solution of 1% of sodium hypochlorite and then washed in the sterile water. After, the specimens were placed in a PDA medium to allow the growth of the fungal, and after the fungal were isolated and the fungal DNA extraction was performed using a DNA kit of extraction. Also, after PCR amplification, the Sanger sequences technology was used to identify the genera of fungal isolates. These are the results about the monitoring activities. So in total, we have found 7 different species. All of them are native. In fact, no exotic species were captured in the area, in particular those of the Xylosandrus genus. This is a really good result. In fact, the species belonging to this genus are now really spread in our country and are, of course, considered invasive for our countries. And we can say also that most of the species belonging to the ambrosia beetles group, and that, how you can see from the graphs, Xylobrinus saxesani was the species most abundant. These are also the results about the type of management. We can see that the number of catches in the whole ground beech forest is higher than the other one, but the statistically doesn't tell us statistically significance difference in the main number of catches between these two habitats. However, there is a significance in the number of catches in the area of we monitored. In fact, the 2 sites in the area of Capo del Morto show a significantly lower mean number of catches compared with the 2 other areas monitored called Capo del Principe and Lecce dei Marzi. Lastly, these are the results about the fungal community sequences. And for this analysis, at the end, we have used just 1 species, the Xylobrinus saxesani, that was the most abundant. And in total, we isolate 260 fungi belonging to 24 different genera of fungi. And we can see that almost the 30% of the abundance can be attributed to the genus Biscognathia, and in particular to the species Biscognathia numularia. This is a really common species of endophyte of the European beech, and in a normal condition, this fungi lives into the plant tissue without development any symptoms. But in a stress condition, actually, this fungi can become a pathogen for the plants. And it may cause cancers, also leaf necrosis, and in general, the decline of the healthy status of the plants. And we also have found other genera with the same habitude, and they are marked with the red asterisk in the table. It's also important to say that Biscognathia is the most isolated genus in both of management types. So, in conclusion, we can say that the fact of no exotic species were found can suggest that the park management strategies could have a positive effect to prevent the introduction, but of course, continuous monitoring is essential to prevent potential future invasion. And we can say also that the results suggest the forest management doesn't seem to influence the presence of these beetles, but further data are needed to confirm this. And also we can confirm with our analysis that ambrosia beetles, the role of the ambrosia beetles has potential vector of pathogenic fungi affecting beech trees. Thank you for your attention.
Thank you very much, Eleonora. Another very good time management from you. We have time for questions. Over there.
Thank you. Nice presentation. And I was curious about the last conclusion about the fungi that could be a threat for the forest. But they are not autochthon. They come with the insect from.
So about
road.
just the fungi? Is
Just
the question?
the fungi. They can be the vector, but of normal fungi that we can find here, or no, or other.
Okay, these fungi that I studied, so the associated fungi that the insect can carry on are autochthon because are in the forest and they spread from one of the other three. But actually there are all other fungi, the symbiotic fungi that carry out inside the insects in the mycanja, that they are allochthon because actually are in the fungi, in the insect, and live actually in the insect. So this insect can bring with them their fungi, but also the fungi that actually are in the forest.
Super cool. Thank you.
One more question here.
Thank you for the nice presentation. I just wanted to ask you if you ever thought about make a comparison between different protected areas levels. You worked on the national park. If you ever thought about working a regional park, or maybe make a comparison between different zones of the national park. Thank you.
Thank you. Yes, actually in the Abruzzo National Park there are zonaging strategies, but we decide to consider just the management because it's strange in that national park because maybe in the B zoning strategies area, there are some old ground beech forest, but also in the A zoning area, so we decide to remain with the old ground beech forest and management beech forest, just to have the data more comparable. But we also have doing another study in a regional park, in the Bracciano Regional Park, in the Oriolo Romano Faggeta. It's actually an old ground beech forest of the region, but the conditions are really different from that one, and actually there we have found a lot of exotic species. So we think that the management can influence the presence of these species, but of course in Abruzzo National Park there are really good natural conditions, so is this the really reason of the maybe absence?
Fantastic. Thank you very much, Eleonora, and congratulations for your nice research. Now we move to the third speaker, Filippo Drigo, who's playing at home. He's from the University of Padua. Filippo.
Good morning. I'm a third-year PhD student, so I take the opportunity for this presentation to at least summarize the 3 years of my work, which is focused on study the photosynthetic biodiversity of the Venice Lagoon with different methodologies, moving from morphology, physiology, and also molecular techniques. Our first aim was to obtain a picture of the species that live in this peculiar environment, and we also wanted to study the gene expression profile and physiological responses of selected species to heat stress. Considering the first part, we started with the creation of a baseline. We made the bibliographic research starting from the 19th century to nowadays in order to obtain records of species that had been found in the Venice Lagoon. And the picture you see on the left represents all the species till the rank of the family that have been recorded, while in the table you can see the number of species that had been recorded for each century. We tried to use this data also to obtain an indication on the ecological status of the Venice Lagoon throughout the centuries, and we did this with the Macroalgae Quality Index and the Ecological Evaluation Index, which categorizes species based on their ecological valence. And the MACI does so by assigning to the highest ecological valence species a score of 2, the green bar, while the Ecological Evaluation Index assigned to those species in the group of the ecological status group 1. And in both cases, we can see that there is a decline of these species throughout the centuries. So we can at least hypothesize that this is also a parameter to obtain to understand that there is a decline of the ecological status of the Venice Lagoon. This and other also information are available are part of a review that we published last December in Frontiers in Marine Sciences. This is a baseline for the monitoring activity we started in 2023, and we continued in 2024 with the seasonal sampling in the 3 sectors of the lagoon in spring, summer, and autumn, in monitoring wood poles for macroalgae and salt marshes for halophytes mainly. And all the samplings have been georeferenced, and also the samples are categorized, and we took picture of most of them to then also upload this data and their identity on the geoportal of the center. For now, we identified these species for each taxa, and for now we found 5 non-indigenous species of the 34 that have been reported in the Venice Lagoon. And the identification have been carried out with both a morphological approach, so using the dichotomous keys, but also using DNA barcoding. And here are reported the different markers we use for the different taxa. Since for algae and plants in general, for algae mainly, it's very difficult to obtain also a good amplification and also have universal marker. This work is still ongoing, and as you can see from the table, even though we can extract DNA from all the samples quite well, the amplification is still tricky, and we are still in the final phases of extracting all the samples we collected, which is almost around 300, and in the next months we will finish also this part. Considering the second part of the project, we focused our attention on 2 halophyte species, which are Sporobolus maritimus and Sporobolus anglicus. These species inhabit the margin of salt marshes, and with their root system, they help to stabilize, to prevent the erosion of these ecosystems. And we selected these species because one is native, while the second one is invasive. And another interesting fact is that the invasive species, Sporobolus anglicus, derives from Sporobolus maritimus in a way that it has doubled the genome size of its parental, of the parental species. And we exposed them to a laboratory, to a heat wave in laboratory controlled conditions, and we monitored the response with different methods. We firstly monitored the photosynthetic efficiency throughout the experiment, and also evaluated the pigment content variation and also the photosynthetic membrane organization. Then we also measured the antioxidant enzyme activity and also evaluated the changes in metabolites present in the leaves, and also with the RNA sequencing technology evaluated the gene expression profiling during the experiment. The experimental design was as follows. So we collected the species from the field and acclimated them for 10 days at 25 degrees during the day and 23 degrees during the night. We then increased the temperature to 38 degrees during the day and 34 degrees during the night, sampling the T0 before the increase in temperature, and then T1 after 12 hours of exposure, T2 after 36 hours of exposure, and then T3 after 5 days. We then gradually decreased the temperature to see if there is a recovery, and sampling at 2 different time points. And for the whole experiment, we had a light-dark cycle of 16 and 8 hours. The first result is the survival. You can see that the native organism, Probolus maritimus, which is represented from the circles, decline after the end of the heat wave, reaching a mortality of 75% at the end of the experiment, while the non-indigenous species didn't die almost. Only one shoot died during the experiment. So it seems that it is more tolerant compared to the native organism. Considering instead the photosynthetic efficiency, and here I'm showing you only the FVUM over FM, we can see that the native organism has a gradual decline of this parameter, which has optimal values of around 0.8, but is species specific. And it has a decline till T4, while the apparent increase at T5 is mainly due to the fact that those measurements were taken from green leaves that were originating from yellow dead shoots. And in fact, after some days, also those leaves died completely. While if we look at the non-indigenous species, we can see that there is a decline, but there also is a recovery. And in particular, if you see the lowest value reached by the non-indigenous species, you can see that it is above 0.6, while in the case of the native organism is around 0.6 or lower. So also there is more stress in the native organism. And these data are also confirmed by the transmission electron microscopy, so the photosynthetic membrane organization of the chloroplast. And here you can see the one of the native organism. You can see the outer membrane of the chloroplast that is completely disrupted, together with the lamella, and also the grana are not so electron dense as they should, and also there are a lot of plastoglobuli that are an index of stress. While in the case of the non-indigenous species, the outer membrane of the chloroplast is still intact. There are some signs of stress, like the distension, the white spots you can see inside. The grana are not so evident, but the lamella, you can see that they are not disrupted as the native organism. And at the end of the recovery phase, this is an overview of the native organism. Zooming in, you do not recognize a healthy chloroplast, while in the case of the non-indigenous species, the grana and the lamella are more defined compared to the end of the heat wave, and there are still some signs of stress, but the recovery occurred. Considering the antioxidant enzyme activity, here I'm showing you only the ascorbate peroxidase, which had a peculiar behavior in the case of the native organism, since we have a peak of activity after the end of the heat wave, while in the case of the non-indigenous species, we have an expected behavior. We have a gradual increase till the end of the heat wave and then a decrease. This behavior will probably be clarified by the analysis of the RNA sequencing data, which derive from 36 biological replicates that have been sequenced with the Illumina sequencing technology. And here you can see the PCA of the gene expression profile of the different replicates. And you can see that the replicates collected during the stress phase, T1, T2, and T3, clusterized together compared to the other replicates that were exposed to lower temperatures, while in the case of the non-indigenous species, we can see the same pattern, and we can also see that the T0 is somewhat separated from T4 and T5, so there could be an acclimatization of the non-indigenous species after the heat wave. Overall, we found more than 20,000 differentially expressed genes in the case of the native organism, and for 75% of these, we had a gene ontology term associated, so we know the function, and we will be able to interpret the variation during the experiment. In the case of the non-indigenous species, we have little less than 17,000 genes, and we are still working on the geoterms associated to these. To conclude, regarding the photosynthetic biodiversity part, so we, for now, used only BLAST as the reference database for the species identification with markers, and in the next months I will also try to use other databases to see if there are some discrepancies or they have data that are not present on BLAST, and also try to use some methods to better distinguish cryptic species and closely related organisms with sequence-based methods for species delineation like the GMYC, but I'm still studying that, so I don't know if I will be able to use it. And regarding the stress response analysis, we are in the final phases on the interpretation also of the metabolomic data and the gene expression, so also in the next months we will also be able to prepare a publication regarding this experiment. And with this, I thank you for the attention.
Thank you very much, Filippo. Very interesting presentation. We have time for a couple of questions. He was able to stay in time here. Two questions. Lady first.
I go first? Okay. Thank you for the interesting presentation. I wanted to ask between the two species, just a curiosity, if there's any ecological competition or they just shared.
They have some differences. At least we see that the non-indigenous species inhabit the lower parts of the salt marshes, the one more covered by tides, while we find the native organism a little bit higher, but they are not so frequent in the same, sometimes we find salt marshes with both, sometimes with only one species. And also regarding their reproduction, the native organisms reproduce, for what I found in literature, mainly with sexual reproduction, so pollen dispersion, while the non-indigenous species mainly stolonification. So also that is part of the distribution. In the next months, we plan to monitor these two species all over the lagoon and see also try to also do a population genetics analysis, and we will see better regarding also the distribution inside the salt marsh.
Thank you.
Yeah, thank you very much for the nice presentation. And I was wondering, just a curiosity, in the first part of the presentation, you showed us that the quality and quantity of photosynthetic biodiversity is decreasing. And could you elaborate, if you know, what are the main factors contributing to this?
The factors are surely the human impact in the Venice Lagoon, which is started in the 16th century. So I would say that is a part. And also the agricultural activity with the industrialization that occurred at the end, around the 19th century. So those aspects surely impacted had an effect of the decline of more ecological relevant species. And also during the 900s, also the high use of contaminants that made the Venice Lagoon eutrophic. Now the situation is better, but it's with the presence of the city of Venice right in the middle, and also Chioggia in the southern part. It's very difficult to return to those conditions, in my opinion. The only most natural part that is still present is the northern part, but also there is a lot of fishing activities. So going back to those conditions is impossible. We can try at least to maintain this biodiversity level, at least for now.
Great. Thank you very much, Filippo, and good luck for your future research. We move to the fourth speaker, Giovanni Felici, also from the University of Padua. Giovanni.
Okay. Good morning, everyone. I'm a third-year PhD student from the University of Padua, from the Department of Environmental Engineering, even though I have a background in economics and sustainable development. My research project actually focuses on, can you hear me? Actually, my research project focuses on evaluating the impact of commercial and industrial activities on biodiversity and ecosystem services. So I'm not going to present my research topic, but a study that we conducted with some colleagues, some GIS expert colleagues from the Department of Environmental Engineering of the University of Padua. So this is the outline of the presentation of today. I'm briefly going to the context of how much it is impactful land use changes. After that, I will briefly introduce the case study, so the company that we are studying in this study, and the research questions, of course. Then I will briefly explain the GIS methodology, the GIS results, and I will conclude with some future developments. So we all know that land use changes is really impactful in terms of biodiversity. Indeed, it's one of the 6 already crossed planetary boundaries. And also according to the IPBS, the Intergovernmental Panel on Biodiversity and Ecosystem Services, it is the most impactful driver on biodiversity. It's one of the 5 main drivers on biodiversity, is the most impactful. So it's something that we must really care about it. So here are just some data about the focus on land use changes. So Veneto, and especially Pianura Padana, is one of the most oil-consumed land. We have a rate of almost 12% in the Veneto region of land converted, quite higher compared to the national and European average. And if we focus on the retail sector, so the supermarket and logistics, the 60% of the total land converted in the period from 2006 and 2023 is accounting for almost 6%. So I didn't want to put the logo, but here it is. The focus is on a case study of a supermarket company that is present here in Veneto. Actually, is the most present in Veneto. You can see that there are 119 supermarkets, 114 are in Veneto, so we decided to focus only on the supermarkets, the points of sale on Veneto. So the research questions was to understand how much land has been converted over time due to supermarket expansion, so the fact of constructing a point of sale, new points of sale, which types of land has been converted, and also a comparison between the GIS results and the non-financial disclosure of the company. So has the company included land use changes in its sustainability reporting, and should the company integrate land use changing in their sustainable development strategies? So this is more or less the methodology that we applied. So we identified and mapping every 114 points of sales. We created a vector layer of the special occupation for each point of sale, so we can know the exact amount of land occupied by the points of sale of Ali Supermercati. we put satellite images and historical area photograph were selected and georeferenced in order to see what there was before the construction of the supermarket. And after that, we did a photo interpretation of the point of sale in order to understand which type of land they converted. So we use QGIS and Google Earth Pro and the regional area photo catalog for the aerial images. So this is just an example of how we operated. So on the left side, you can see that on the left side of the left box, there is the example of one point of sale at 2024. So going back in time with the satellite images or aerial photographs, we could have seen if there has been conversion of land or not. So on the left side, we can see that there has not been conversion, while on the right, there has been. So we distincted these two categories of land conversion. So for the one that occurred land conversion, we decided to divide in 8 different land use, land cover classes that are reported in the bottom of the slide in order to understand which type of land has been converted. So these are the results. So out of 114, 50, so more or less the half of them had converted land, so the conversion from rural to urban land. 32 were built on an already existing urban area, as we saw in the previous slides. And for 32, that is quite a big limitation of the study, for 32 was not possible to determine if there has been land conversion or not, because, for instance, if you look at, a supermarket that is on a ground floor of a residential building, you cannot know when it was opened, the supermarket, or not. Of course, we sent an email, several emails actually, to the companies in order to have more information, but we did not have a reply. So if we look at the percentage in terms of hectares of land, actually rural area is quite impactful because, as you can see, 62.7 hectares has been converted out of 95, so almost 2/3 of the total land cover of the points of sale. And also, I would like also to point out that maybe for the people that live in Padua, you know that there was a debate about the construction of a new logistic hub in Camin that actually cover 15 hectares of rural land, so more or less 1/4 of the total converted land. So also, if we look at the time distribution of the points of sale that has been constructed, we can see that we could expect actually a decrease in the land conversion, but if you look at the last 15 years, 16 out of 23 of the new point of sale were built on a rural area. And in terms of hectares, they converted 73.7, almost 3/4 of the total land cover. So these are the results of the land converted. Just I will go briefly on that. You can see that almost half of actually almost 3/4 of the converted land was agricultural or herbaceous area that are of course more important in terms of urban biodiversity and mitigating heat waves. And almost the half of the total land covered was rural or semi-natural land. So this was a screenshot of their non-financial disclosures, so their sustainability reporting. So for the one that don't know, every company with more than 500 employees and I think 2 million of net profit per year must report their social and environmental impacts according to EU Directive of 2014. But as you can see, the main focus of the sustainability reporting focuses on CO2 emission savings, clean energy and recycling, but there is no mention about land take and land consumption. Also, if we look at the compensation project, we can see that the almost 50,000 trees that they say that they have planted directly by the company, they were not put directly by the company through customer driver initiatives where loyalty points could be used either to plant a tree or redeem other rewards. So the decision of planting a tree is on the customer side and not on the company strategy side. We also proven that the 23.5 hectares of land that they say that they converted from urban to rural, there was no land conversion because we saw that through a GIS analysis that they plant trees where there was already a forest or a rural area. And we also saw through the spatial analysis that the proximity between afforestation sites and the point of sale on converted land revealed an average distance of 40 kilometers, so quite far away where the land conversion occurred. So coming to the end, we believe, I mean, we didn't want to attack directly this company, but it was, I mean, we had data on these, so we wanted to show that companies like Bali, but many others, should include land consumption in their sustainability reports. It's really quite important, and the EU regulation doesn't mention it. There is no strict regulation for checking what the companies, the claims of the company. So there is no jurisdiction on checking every declarations on the companies, and it's really important. It's something that we have to think about it. So we need, there is a need for greater clarity in green claims to prevent greenwashing or misconceptions. And finally, we would like to apply this methodology to other case studies. With a talk that we have with Professor, it would be interesting to see also other sectors in the logistic and in the retail sector in order to understand how other companies work in this field. And in September, I'm going to Wageningen in the Netherlands, and we would like to check with this kind of methodology if the companies that are getting some environmental certification, like for instance the free trade, if they effectively, I don't know, if they didn't consume land for getting the certifications. So thank you for your attention. Here's my email and my LinkedIn. And here are the references. Thank you.
Thank you very much, Giovanni, for this very interesting point of view. And we have time for a couple of questions. Here one.
Thank you. It was very interesting to me. And I was wondering, I don't know if you investigated also in this sense, is this land consumption somehow supported by some reasons, like new development, residential developments, or new infrastructures, because usually this kind of logistics hubs are supported by new highways or railway infrastructure. Maybe, I don't know if you also investigated in this sense, but thank you, it was very interesting.
No, really good question, really good point, because for me it's really quite interesting. Yes, unfortunately we cannot prove this, but we have some hypotheses that this company has how can I say, a residential company where they built effectively residential buildings. And also, if you see in most of the point of sale that where has been occurred land conversion, you can see that also some residential buildings came up next to the point of sale. But unfortunately, we cannot prove that was like, I mean, we should go directly to every 114 point of sale and seeing with our eyes if the residential building was from the Ali companies or not. But yes, unfortunately, we cannot put this data on the paper. Thank you.
Any other question? Here, Andrea.
Land consumption due to construction of roundabouts along roads, because as a person living in Veneto, one of the most visible effects seems to be that they build these roundabouts everywhere at any crossroad.
I think I didn't get the question, sorry. I'm sorry?
Rotatorie stradali.
Ah, okay, okay.
And they are usually in agricultural environments, so?
Yes, exactly. Now, of course, I couldn't show every 114 point of sale, but in most of the point of sale where there has been land conversion, they also added street and roundabouts. And yes, I mean, this is also a limitation of the study because the limit of the perimeter that we included into the data. So yes, with Professor, we speak about it yesterday. Maybe the data is overestimated because sometimes we included roundabouts or parkings, or for instance, if the supermarket is in a commercial center, I mean, which is the limit of the supermarket or not? But yes, absolutely, this is quite a big limit, but it's a photo interpretation. Thank you.
Thank you very much, Giovanni. Very interesting, but times is over. And we move to the fifth speaker, Francesco Ferraro, also from the University of Padua.
Hi everyone, good morning. I'm Francesco, I'm a PhD candidate in physics here at the University of Padua. And today I will present our latest work, which is titled Time Delays Induce Synchronization and Chaos in Complex Ecological Communities. So the main topic of my PhD research is understand quantitatively how to model ecological communities. This is, in general, very hard task since communities are very complicated. And this is because in communities there are many different species that are interacting, and there are multiple environmental factors that act upon them. So it is not trivial to understand which features have to be included in models and which features are relevant. One of the features that can be introduced in models is time delays. Time delays essentially are introduced to model the fact that in ecological communities, what happens to a species does not instantly influence other species in the community, but it does so after a certain time delay. What we were able to show in our work is that delays arise very naturally when the dynamics of the community is coupled to an environmental feedback. So, for example, you can imagine observing the dynamics of a community, for example, a community of birds, which is influenced by some external variables, and maybe the dynamics of the full ecosystem, so the community plus the environmental variables, is not delayed. But in the moment you observe only the dynamics on the space of the community variables, so you project these dynamics onto this lower dimensional space, then it appears that the dynamics of the community is delayed. And apart from this way that we show that it's very well known in physics, this appearance of delayed effects in equations, delay naturally arise, for example, to some very known ecological processes, such as maturation times. For example, some species take some time to develop into full maturity, so that gives naturally a delay effect. Also, if the community is feeding on resources which have a long renewal period, but delays can also appear due to seasonality. So, given that, in general, as we show in our work and from general arguments from there are many ecological mechanisms that can give rise to time delays, it is natural to ask the question, when are they important? In the sense, when their effect is relevant in community dynamics. So to understand when they are relevant and when they are not, what we did was studied the central model of community ecology, which is the Lotka-Volterra equation, which you might know from the predator-prey Lotka-Volterra equation. And this is just the generalized version, which includes an arbitrary number of species. In this equation, xi is, for example, the biomass density of a certain species. And this equation basically sets the growth rate of species I as a function of all the abundances of both species I and all the other species J in the community. And as you can see, this interaction is mediated by some interaction matrix, alpha ij. And as you can see from the model, we included explicitly a time delay. So what happens at time t to species I is influenced to the state of the species J at times t minus tau. So this tau is what is called a time delay. And what we did was study the properties, the dynamical properties of this equation as a function of key parameters of the interaction matrix. And what we found is that we studied the properties of the dynamics as function of the interaction mean of the interaction matrix and of the interaction heterogeneity in the community. So moving horizontally in this graph, basically we are setting the competitiveness or cooperativeness of the community. So moving to the right, the community is more cooperative, while moving to the left is more competitive. While moving vertically sets the heterogeneity in the interaction, so how much a community is diverse. And as you can see, there is a transition from a phase of stability, which is the pink one, towards a phase of oscillations, which is the orange one. And so there is a threshold of competitiveness above which the community displays synchronization. So what you observe in the community is that the species abundances start to oscillate with the same frequency and a very small space displacement, and which is what we term synchronization. So our result is that when a community has sufficient competition in it, then it might present synchronized oscillation, and in that case, time delays can be relevant. We also analyzed a community with some structure interactions. And specifically, what we considered was a community in which predator-prey pairs are present. And in this case, we show that we have the same phase, the orange phase of synchronized oscillations, but we also have an interesting phase in which there is a phase of delay-induced chaos. So in this phase, the abundances of the community start oscillating with irregular motion. And the dynamics is very sensitive to small initial differences in initial conditions. So to sum up, what we showed in this work is that time delays are not ad hoc mathematical element, but are inevitable in models of community ecology. But we were able to show when they are important and when they are not. What we show is that in competitive communities, time delays effects can induce a phase of synchronization, while in predator-prey communities, they can induce both synchronization and chaos. And all this framework might seem very theoretical, and indeed it is. We have no comparison to experimental data. But this whole framework is in principle experimental testable in microbial communities where it is possible by changing, for example, the pH and increasing or decreasing the number of species. It's possible to change both the average interaction in the community and the heterogeneity. And yeah, that's all I wanted to say. You can find the preprint at the link on the screen. And thank you for your question, for your attention. I will take any question.
Thank you very much, Francesco. Very good use of your time. So we have plenty of time for question. One there.
Hi, and thank you for the presentation. It's very interesting. Actually, I do a lot of time series analysis, and I'm confident with time lag, so you call it time delay. And it's interesting, like a lot of people always neglect this variable, but it's nice to see this project. Now, my question is, the time lag, the time delay in your model is somehow arbitrary, right? You choose it, and are we talking about minutes, hours, days? How do you choose it?
Well, depends. Could be any could be any time delay. Essentially, what I can tell you is that what you have to consider is the ratio between the time delay that you put in your system and the intrinsic growth rate of species. Essentially, what we can show is that when they are comparable, in a sense, they are on the same order of magnitude, then it is relevant. If the delay is much shorter than the intrinsic growth rate, then essentially it is, you can also not consider them. If I want, the ratio is pi over alpha.
More question? One more question here. Oh, okay, 2 more questions.
Thank you for your presentation. I just wanted to know if you have plans to try it out in a predator-prey setting. Do you have any plans to push it to the experimental part?
Well, at the moment, no. But I think this work builds upon a recent trend of trying to make quantitative statements in ecology. And I think the current platform, which is the most promising to test theory, is that of microbial communities. Like, a lot of work has been going on in trying to assess whether these models are enough to capture the dynamics of microbial communities and when they are not able to do so. An important element that has to be addressed before moving to experiments is to include the effect of noise. All models that I've showed are deterministic in the sense that what happens at time 0 exactly determines what happens after future time, but including noise is something that has to be addressed before moving to experiment. But yes, in principle, it can be tested in experiment.
There is another question there.
I wondered whether there was a reason why you put the time delay in the competition or predation part, but not in the growth rate component.
Well, if you are saying why we put x dot equal to xi both at time t, that is very natural to assume that the species grows proportionally to the current population. So that is, I mean, you can also devise a model in which that is not the case, but is not what is usually assumed, just because of the way population grow. If you're asking why in the regulation term we put the delay only in the competition part or cooperative part and not in the self-interaction part, I didn't show it, but we studied in more detail what happens when also self-interaction are delayed. And yeah, you can find all the details. I mean, we can chat, but you can find all the details on the preprint.
Time for another quick question. Okay, no more questions. Thank you very much, Francesco. And congratulations for your research as well. Now we move to the next speaker, Simon Masson from the University of Turin.
Good morning, everyone. So I'm Simon Masson from the University of Turin, and I'm glad to present you a part of our result I've done during my PhD. Today I will speak about urban parks, and more particularly urban lawns and their associated ecosystem services. So, as you probably know, about 70% of the human population will live in cities by 2050, according to recent estimations. Cities could have many urban green space, as you can see on this scheme I extracted from Sun and Hall, which can have many benefits and ecosystem services, such as mitigating flood events, or heat island effect, or even sequester pollutants, carbon dioxide. They could also have an impact on mental health by favorizing, for example, the practicing of sport, or also directly with their biodiversity by promoting the immunity system, for example. So here, the aim of the study was to see the impact of the age of urban lawns on diverse ecosystem services along a national latitudinal gradient. So 5 Italian cities were considered: Milan, Turin, Florence, Rome, and Naples. And the sampling was done in 2023 between late spring and summer. In each city, we consider 3 recent parks with less than 20 years establishment, and 3 historical ones, and a comparison with 1 less disturbed site as a control. In each park, we have done 3 plots of 16 square meters, and in each of the plots, we sampled 3 topsoils that we pulled together. So the quantitative variables analyzed were numerous. So with the soils, we assessed soil physicochemical features, enzymatic activities, the microbiota with metabarcoding approach, and we also, with the help of AirSAF, have done a QBS arthropod index, and a vegetation survey was done thanks to all the local partners. So here you can see all the ecosystem services we assessed. For example, we have chemical ones with the nutrient availability, or more physical one with soil porosity, or even biological one with, for example, pest control, which take into account the relative abundance of animal and plant pathogenic fungi. Here you can see that we pull one or more ecosystem function together into an ecosystem services. And after we've done the mean of all these ecosystem services to obtain the ecosystem multi-service indexes. Before doing the calculation, all the values were standardized thanks to this formula of Wang et al. Let's move to the results. So we can see, if maybe I will try to guide you. In a less urbanized site, there is a higher organic matter decomposition, and there is also a higher soil porosity than in urban parks. Moreover, there is a higher carbon sequestration here than in recent established park, or a higher soil biological quality than in historical lawns. If we only compared urban parks, we can see that net primary productivity was higher and pest control too here in historical lawns than in recent ones. Here we have the result for the ecosystem multi-service indexes, so the mean of all the ecosystem services I've shown before. And we clearly see the trend in favor of age, with control area better than historical, better than recent. Here you have only the quantitative, so we also have done an analysis on qualitative, and in order to do that, we've performed a threshold analysis. So we can see that the number of ecosystem services that reach 50% of functionality is higher in control site compared to urban parks, and for 75% we have the same trend than for the ecosystem multi-service index. We also looked 2 other indexes, which are multidiversity and multidissimilarity. For the multidiversity, we see that it's higher in control areas, and the multidissimilarity too. In fact, if we go into detail, we can see that there is an homogenization of communities with the dissimilarity for the vegetation or for the bacterial communities. And however, for the diversity, it's only significant for the fungal communities. Finally, we also done structural equation modeling thanks to my period abroad in Spain. It allows to see direct and indirect interaction between environmental variables on this ecosystem multi-service index. We can see that latitude, so the city, has a direct positive impact on precipitation level and the two soil parameters, so pH and percentage of clay. Moreover, mean annual precipitation has negative direct impact on both and H2 on both soil parameters. And finally, historical loans and percentage of clay have a direct positive influence of ecosystem multi-service index. Finally, we also done a focus on microbial diversity, which is what we are studying in our lab. And we can see that some organisms discriminate well between both age of park. For example, Nocardoides and Microvilga bacterial genera well discriminate recent from historical ones. And we have also more pathogenic genera in recent parks and more saprotrophic fungi in historical lawns. Finally, as I've shown before with the dissimilarity, we can see an homogenization of bacterial communities across cities and park age with, for example, the same relative abundance of phyla in the city of Rome. And it's the same for fungal classes, but with more distinct communities across the cities. So to conclude, we have observed that age shapes ecosystem functions in urban lawns, that older urban lawns support greater ecosystem services, and there is a global homogenization in Italy of soil microbial urban biodiversity. But we can observe some microbial genera associated to recent or historical lawns with the left analysis. So thanks to my tutor, my colleagues, all our partners, and you for your attention. Thank you.
Thank you very much, Simone, also for staying in time, and we have time for a few questions. Questions? Here, one question.
Just a curiosity, why do you think clay is positively correlated with the provision of ecosystem services?
I didn't show all the data, but ERSAF in Lombardia have shown that it has a positive impact on arthropod communities, for example. And as arthropod communities are englobed in the ecosystem multi-service index, maybe it could be explained like that.
One more.
Thank you for the presentation. Just one question, because lawns in urban areas also create many disservices for the pests or mosquitoes. Did you have any data about it, or did you notice anything in the process?
For the disservice, we only see the pest control, so the relative abundance of pathogenic fungi, but we didn't assess other negative impact of urban lawns.
More question? Simone, would you be able to provide some suggestions in newly established urban lawns to increase the ecosystem service they provide?
Like that, the very difficult question. Considering the parameters, I think, yes, that we could, for example, put flora meadows, but because we didn't look at them, but for bringing new pollinators or reducing also pathogenic, maybe. I don't know other things.
Anyone else? Okay, so thank you very much, Simone, and congratulations for your research as well. Now it's time for Ate Ndiforngu from the University of Padua. Ate, floor is yours.
Good morning or afternoon, everyone. I'm Ndifongo Ate from the University of Padua, and I'll be presenting part of our work that we carried out with our partners, partner companies, and research labs. So I'll be talking about monitoring from the point of view of waste management, and a contaminant of concern would be the per and polyfluoroalkyl substances. Thank you, sorry. Okay, so we'll be talking about the per and polyfluoroalkyl substances in the end-of-waste plastics. And just to give some background, these substances are popularly known as PFAS, and they are very interesting because they are synthetic, they're man-made chemicals, which have excellent properties, excellent non-stick properties, so they are used for a variety of products that we encounter daily. They are used for food packaging, they are used for textiles, they are used for cosmetics. For the ladies, your mascara and your foundation have PFAS. They are also used for automobile and aerospace, just to name a few. And of recent, they have become very interesting because they have been associated with health concerns such as endocrine disruption, hormonal disruption, and some cancers. So if we look at the photo on the left side, the entire PFAS life cycle subjects the environment, thus biodiversity, to a plethora of contamination from PFAS. And the sector that I want to focus on is the waste management sector, of course. And in this sector, circular economy demands that since we are transitioning from linear, we need to incorporate a lot of recycling, and it's problematic for us because PFAS are created for different reasons, and so they have different life cycles. So when they come to the waste treatment plants, the recycling plants, it's difficult to treat them as a homogeneous substance, and so the tendency is that they are not usually removed totally from the treatment. And so the EU has recommended some limits for recycled materials, so 25 ppb for singular PFAS and 250 for the sum of PFAS from target analysis, and 50 part per million for total fluorine, which is a good indicator of PFAS. And in order to understand the presence of PFAS in waste, because we know in plastic waste, it's popularly known that PFAS is a part of plastic waste, so we decided to conduct a review on the presence of PFAS from plastic waste, and our study showed us that much of what was found in the studies were actually below the limit of quantification, which is supposed to be a good thing, but we observed that most of the work that was carried out had diverse protocols used. And so it was difficult to really reliably say that the results of these reviews are safe, in quotes. And also, since we are interested in the impact that it has on health and biodiversity, of course, we had an eye on studies that gave migration potential, and there were very few. In order to bridge this gap, we decided to conduct our own research to monitor the presence of PFAS in recycled plastics, that's using different standards and different protocols, and we also wanted to characterize the migration potential into 3 different exposure routes: food contact, oral or mouthing, and lastly, dermal exposure. So the first thing we did, we contacted some companies. We got samples from 2 recycling companies and 1 recompounding company, and there were over 37 samples of plastics from different kinds of polymers, polypropylene, polyethylene, polyamide, etc. And all of this came from either pre or post-consumer goods. And the sectors that were involved that volunteered to give us samples were automotive, the furnitures, building, and good distribution. And depending on whether they came from recycling or recompounding, they were either fragments or in pellets. Our first method was the target analysis. Target means that we had a fixed amount of chemicals that we wanted to observe. We used the liquid chromatography mass spectrometry method, and for this we used two extraction solvents which are popularly used by local companies to analyze PFAS in waste. The first one was using methanol extraction. Methanol is used for, it's a protocol that is used for the quantification of PFAS in plastic containers, and the second, tetrahydrofuran, is used for the quantification of phthalates in consumer goods. And then after that, we carried out the total fluorine, which is a non-target. Since PFAS is per and polyfluoro, the fluorine part, so the non-target was to be able to quantify the fluorine that is in the samples. And then for the ones that, for the samples that we think that will not have PFAS but could have chlorine, we decided to confirm it with a TOP assay. It usually identifies the hidden PFAS, and so we wanted to confirm it with the TOP, and lastly we did a triplicate just to be sure. Now the first part of our results, using the two different extractions, first of all, we can notice a big difference. Or let me start with the fact that we expected that since there were 40 types of polymers that we aimed at, we thought that maybe we would have a significant number, but no. Only 6 types of polymers were identified using the LCMS, notably PFBS, PFBA, as you can see on the board. And we also noticed that depending on the extraction protocols, the tetrahydrofuran extraction significantly extracted a large part of a large quantity of the polymers, and most of them, more than 87% for the tetrahydrofuran, were above the LOQ. Meanwhile, the med extraction was significantly lesser, and much of them were below. And so this could be an indicator that probably the tetrahydrofuran extraction is more accurate than the med extraction. Also, we found that for the med extraction, just 3 polymers were above the fluoroteloma, PFBS, and PFOA. The results for a total fluorine, the non-target analysis, showed us that all the samples had PFAS as compared to the previous one. Every sample had, not PFAS, fluorine. So all the samples had fluorine inside. They contained fluorine, and most of them were about 43% were above the recommended limit by the EU, which I presented before, which was 50 parts per million. Most of them were above, a good part of them were above, but all of them were greater than the sum of the target analysis from the THF and the methanol extraction. However, since total fluorine is composed both of organic and inorganic fluorine, it will be misleading to completely rely on the results from the total fluorine, and so we decided to understand if talc was present in these samples. Now, talc it's a plastic filler that is, sometimes it contains impurities such as inorganic fluorine. So we thought if we identify talc in these samples, then possibly there's inorganic fluorine also in the samples. And yeah, we conducted it on several samples, and we realized that yes, there's a presence of inorganic fluorine. The second part of our results, the migration tests, we conducted 3 of them, but for now, I have the results just for the oral exposure. And the oral exposure showed us that only one PFAS, PFBS, was detected migrated from the samples into the solvent, and it was significantly within the limit, so it's safe. But then PFBS has been known to be highly soluble in water-based, it's very water soluble, and so it's just an eye-opener that we should look out for this particular PFAS in stuff that have to do with oral exposure. In conclusion, we conducted both target and non-target analysis for PFAS in plastic, recycled plastic samples, and overall, tetrahydrofuran has a higher extraction potential as compared to methanol, and the TOP assays, they didn't show us any significant difference, so it confirmed what we found. Also, the fluorine values that we found suggest that the target analysis may be very useful also. The non-target may also be very useful in order to have a better holistic understanding of the PFAS contamination in recycled plastics. But then, since the presence of talc was also shown, we cannot completely rely on that. And lastly, PFBS can migrate into saliva. Thank you.
Thank you very much, Ate. We have time for questions. Questions? Don't be shy. Ate, do you have any data on the impact on organisms of these contaminants?
Of PFAS?
Yeah.
Not yet. I have for microplastics, yes, but not for PFAS.
Okay. So you're looking for those contaminants on organisms or in the environment?
So my research is really quite large because I started with the impact on soil and soil organisms. I'm conducting the rhizo test to understand the migration of microplastics into plants. And also, since microplastics adsorb to contaminants such as PFAS, they carry it along, it's also one of our perspectives, but I have for now only those for the impact on plants, on soil organisms, on humans from in vitro analysis, but not yet for PFAS.
Okay, thank you. We have time for one more question, if there is any. Okay, so we thank Ate for her very nice research. And we move to the last speaker, I believe, who is Cristina Ricucci. Is she here? From the University of Modena and Reggio Emilia, right?
Okay. Good morning, everyone. In this contribution, I'm going to present the Lago Lao case study as an example of the multidisciplinary approach adopted within my PhD project that is focused on palynological research to investigate the Holocene plant diversity changes within 3 Italian natural areas. Understanding the long-term ecological trajectories and plant diversity responses to climate variability and human pressure is crucial for contextualizing the ongoing climate change and informing conservation strategies that account for ecosystems, resilience, and vulnerability. In this context, palynological research is a powerful and useful tool, providing insights into the climate-human-nature relationship on a long-time perspective. The study of these complex dynamics within a broad-scale pattern requires detailed and accurate local data within a wider spatial distribution. So in this context and framework, Lago Lao site is a high-altitude Sphanium pit bog in the Ligurian Alps Regional Natural Park. And despite this is considered a priority habitat in Europe, the site is not currently included in the Natura 2000 network. We selected this area for three main reasons. Because Southwestern Alps are biodiversity rich and climate sensitive areas, and then because they have a long history of human presence, dating back to the Pleistocene. And finally, for the lack of paleoenvironmental records with a robust chronology covering this time period in the study area. As I mentioned before, we adopted a multidisciplinary approach involving a sedimentary description, radiocarbon dating, and the age depth model elaboration performed using the BCron, R package, and INTCAL20 calibration curve. And then subsampling, chemical treatment, light microscope, palynological analysis, and pollen percentage diagrams and cluster analysis elaborations with TILIA software. These paleoenvironmental data were then integrated and compared with the archaeological documentation from the study area and with independent paleoclimatic data set at global, regional, and local scale. In particular, we considered the documented global cooling events and warming periods, the Holocene Mediterranean sea surface temperature anomalies, the last about 5,000 years of North Atlantic oscillations, and finally, the past local annual temperature and precipitation were estimated using the KELSA Trace 21K model, 1 kilometer resolution global climatic model covering the last 21,000 years. So the age depth model from Lago Lao site shows that peat sedimentation started about 11,000 years before present, with a continuous sedimentation until recent times. Instead, palynological analysis reveals high floristic richness with a total of 140 pollen taxa, including about 30 woody plants and 100 herbs. Both paleoenvironmental and paleoclimatic data are reported in this figure, and the pollen percentage diagram on the left includes selected pollen taxa and pollen zooms. And it describes the pollen percentage trend from the lower and older levels to the more superficial and recent ones, and then divided in different pollen zones identified by cluster analysis. So between about 11 and 8,000 years before present, Lago Lao results show the role of increasing warming and moisturing during the Younger Dryas early Holocene transition in driving the succession from cold steppe vegetation and herbaceous peat formation to the first colonization of pioneer cold trees like Betula and Pinus. This first transitioning phase is followed by the progressive expansion of montane Mesophilus broadleaf forest during the Holocene climatic optimum, so under more stable, warmer, and moister conditions. This ecological succession is like a common mark of the late glacial transition, also documented in the Nerbi Maritime Alps and in northern Apennines. Furthermore, during this period, 3 global cooling events occurred, and they aligned with regional and local paleoclimatic data and correspond to arboreal cover decreasing phases, suggesting a probable effect of cooling on local environment. Instead, between about 8 and 3,000 years before present, significant paleoenvironmental and paleoclimatic changes are recorded. In particular, in the mid-Holocene transition is characterized by a sharp warming and moisturing during the Holocene thermal maximum, which may have promoted the Abies first expansion, marking the tree-line raise and the establishment of subalpine forests in the area. Instead, in the mid to late Holocene, a progressive arboreal cover reduction involving both montane and subalpine forests are detected. These environmental changes may be related to climatic and or anthropogenic factors. In fact, during this period, an increasing climatic variability and drier conditions are suggested by the occurrence of global cooling events, by the decreasing trend in Mediterranean sea surface temperature anomalies, the positive now phases, and the reduction of local precipitation. But on the other hand, the opposite trends of arboreal and anthropogenic pollen suggest a probable human contribution. In fact, this period corresponds to the spread of Copper and Bronze Age human civilizations, along with the high-altitude migration and diversification of human activities, including cultivation, fires, mining, and transhumance. Sorry. So over the last about 3,000 years, a progressive arboreal cover reduction culminates with its local decline, the expansion of wet meadows, and an intensification of anthropogenic pollen indicators and cultivated trees during the warmer, drier medieval climatic anomaly. And finally, between the colder Little Ice Age and the warmest 20th century, a slight forest recovery is followed by its further reduction with Abies local decline, wet meadows decrease, and anthropogenic pollen persistence. These environmental changes correspond to marked variations in Mediterranean SST, the highest value of local temperature, and a drop in local precipitation. These results will be implemented with further data elaborations, including the rate of change analysis to estimate the beta diversity variations over time, and with the multivariate analysis to assess the taxa sensitivity to climatic and anthropogenic factors. So these integrations will be useful to quantify their impact on local biodiversity and the long-term responses. But for now, we can summarize the main findings as follows. First of all, the establishment of montane and subalpine forests in the area was probably promoted by the increasing warming and moisturing during the early mid-Holocene. But the climatic instability and past human activities may have influenced their following decline after about 8 and 3,000 years before present. These factors may have also influenced the increasing vulnerability of these forests documented during the last millennia and determining significant changes in forest abundance and composition. So with these findings and this study, we want to underline the importance of reinforcing the altitudinal ecological connectivity, for example, including this area within the Natura 2000 network, in order to prevent or mitigate the impact of climate change on these vulnerable forest ecosystems and peatlands priority habitat. So thanks for your attention.
Thank you so much, Cristina. We have time for a few questions. No question? Cristina, can you detect extinction of species or invasion by new species in your time frame observations?
Invasions, yes, because we have a local data and is enough to estimate the presence of an allochthon species. But the extinction maybe need more data on a broad scale. But yes, it's... And for example, the rate of change analysis is very useful because estimate the rapidity of changes. So it could be a tool to evaluate also the extinction. But
any other question? We have Fabio.
Hi, Christina. Fantastic presentation, and the graph was amazing as well. So, I'm a lay person in this topic, so I just want to check again how this can be applied to ecological restoration. So, if there are any applications, and because it's really amazing, and sometimes, yeah, we don't have historical maps, for example, but with your methods, we can go back like thousands and thousands of years. So, yeah, just this curiosity.
Yeah, I think that it's a key point because sometimes when for example, in the selection of areas within the Natura 2000 network, we consider the current distribution and presence of species. But I think that will be useful to consider also the past, and because it gives us information about the vulnerability of an area and also the potentiality of a territory. And this is called vocation. So I think that it could be useful in this sense. And sometimes we have also examples, for example, from Sape site, that is my third site, is near a mixed silver fir beech forest, and there we see a restoration process after the institution of the Pollino National Park. So it's also a way to monitor the responses and effect of our conservation measures, but also the activity of past human activities that are similar to us.
Fantastic. Other questions?
You can get from your sediments?
It depends, because it depends by the sedimentation rate of the bogs, the peat bogs. In this case, is about an average of 300 years. But sometimes, in other contexts, when we have a slow sedimentation, we can reach higher resolution.
Okay. So if there are no more questions, I think we can close this session now. It's 1 o'clock, so we're perfectly in time. I would like to I would like to thank all those who made a presentation this morning. Fantastic research, very diversified, as we would expect in a group of people who study diversity. And also I would like to thank all those who asked questions, because this helped us to stimulate the discussion and to go deeper into the details of everyone's research. So I think we succeed in having a very nice session, and I believe now it is time for lunch, right? Thank you very much, and again, an applause to those who made the presentations.
Questo lo usi tu?
Cominciare voi? Cominciare voi però.
Sì. Avanti da prima, basta dire che partiamo. Sì.
Hi, everybody. So, welcome back. I hope you're still awake after lunch. Now it's the time of the perspective. We are here together with Francesco Fratti, coordinator of Spoke Free, that you know very well since last years. So now we've been talking for 3 years now, like 2.5 years of NBFC, of how we started, how we proceeded with our project, and now we are approaching, unfortunately, the end of the PNRR. You and we are all aware of this. So we know that a very important topic, very urgent for all of us in this room, is also talk about the legacy of NBFC. So this perspective will be focused in particular on the legacy, or better, the legacies of this center. So Francesco, which do you think could be the legacies of this, of the National Biodiversity Future Center, especially taking in consideration our public today, that are our PhD students that followed us for almost 3 years now?
Okay, first of all, let me say that what you see on the screen is what has become the main claim of the National Biodiversity Future Center. Biodiversity is the solution. In one sense, we have the idea that these 3 years of duration of our research have done something in convincing the people, the citizens, and the policymakers that protecting biodiversity is something that we cannot do, we have to do now. It is not something that we can postpone any longer. And at the same time, we would like to condense within this claim the idea that taking care of biodiversity is not necessary for its own sake, because some environmentally fond people are taking care of that particular tree or that particular animal because it's cute, it's unpolite not to take care of it, and so forth. We believe that biodiversity is a solution for many of the problems, or maybe part of the solutions, for many of the problems that affect humankind in this particular moment. Later on, we will discuss about climate change, for example, which indeed is one of the issues, maybe the most important one, or certainly one of the most important ones in this period of time, at the beginning of this third millennium. But going back to the question, the first thing that occurred to me when I'm thinking about the legacy of the National Biodiversity Future Center is human capital, is you. The reason why we're here today, the reason why at the beginning of the project we set the ambitious goal of creating, of educating a new generation of scientists, which is one of the 5 key topics from the very beginning of the National Biodiversity Future Center, was because we were all aware of the fact that not only biodiversity was eroding, but even the biodiversity experts were declining. There are many collection of data showing that around the world, not only in Italy, biodiversity experts are declining. There are no longer, or we are experiencing an erosion of those who are able to identify a species, those who are specialists of a group, either of plants or animals or microorganisms or fungi. So when we started, we decided that one of the goals of the MBFC should be educating a new generation of scientists, you. So we believe that this is legacy number 1, because the most important thing that we have produced is investing on human capital. This, of course, has been done using several initiatives. One of them is the National PhD program on biodiversity. Just to mention a few numbers, we had 30 fellowships in the first year of the national program and 54 fellowships in the second year, this current academic year, 24-25. But before the National Biodiversity PhD program started, with the resources of the National Biodiversity Feature Center, we had funded over 150 positions in PhD programs across the country. So those around 240 students, PhD students, are probably one of the most important legacy of our program. And this is intangible. It's not an instrument. It's not a product. It's not something that has been sold. This is something that will stay with us and hopefully will be able to guarantee to the country the necessary knowledge to study, to monitor, to preserve, to restore, and to valorize biodiversity.
Thank you, Francesco. I think that there is one other legacy that we may left to the country, because I think that sometimes scientists and researchers may feel alone in communicate the urgency of protecting biodiversity, but I think that the lay public can help us in doing this, and that's why we also took on some program to disseminate biodiversity and the research that you and all the researchers and from the center are doing on biodiversity. So that is another one of the other legacy that we left, maybe.
I agree, and I believe that one other important legacy that we hope we have produced is the impact on the citizens, the impact on the population. And we have been doing this by communicating sciences, and especially biodiversity science, as best as we can, in getting the private sector involved, something which is sometimes incredibly difficult. But when you hear from, for example, the World Economic Forum, that almost 50% of the global GDP depends on nature, maybe it's easier to convince even the private sector that investing money in biodiversity is crucial. And there is another important issue, which is the link between biodiversity and human health, the famous One Health concept. Hella Scena, one of our colleagues, scientific coordinator of one of the spokes of the National Biodiversity Feature Center, invented a sentence which she mentioned during the forum in Milan last week, when she said, investing in biodiversity is like investing in the health system, in the national health system. Because with a healthy environment, we also improve human health. There is another issue that we have to mention: the ability that we have had in getting the citizens involved. The last forum on biodiversity in Milan ended in the Milan Triennale with a sort of forum to which some of our European external reviewers participated, And one of the conclusions of that forum, one of the questions of that forum was, have we been able to induce, to make an impact on the policymakers so that on the basis of the results of the research of the biodiversity, of the Biodiversity Future Center, the policymakers have data to produce regulations which are necessary to protect biodiversity. And one of the answers was, we will be able to convince the policymakers if we convince the citizens on the importance of biodiversity, because the citizens are those who vote those policymakers. I kind of summarized this concept in this slide. The best option we have to convince policymakers that monitoring, preserving, restoring biodiversity is important is to get the citizens involved and thereby convincing them. One of the ways we can get the citizens involved is through citizen science project. Some of you, many of you, most of you, I believe, know what citizen science projects are. Maybe some of you have participated to those citizen science projects. We defined a strategy to let citizen science become one of the corner points of the National Biodiversity Future Center, including establishing a working, starting with a working group on citizen science, which is coordinated by Andrea Sforzi. And within this working group, we have several goals: share good practices, organizing training courses on citizen science. We have had one of those training courses in Siena in November 24. We are building a national platform for citizen science projects so that all those people, all those researchers, all those organizations who are organizing citizen science projects have a place where to put their project, where to communicate the results, where to create links and networks with other projects. Citizen science participated to the MBFC sampling weeks a couple of weeks ago. And finally, we are talking with the ministry to sign an agreement between the National Biodiversity Future Center, the ministry itself, and Citizen Science Italy, which is the association for citizen science in Italy, which is in turn linked to the European Association for Citizen Science, with the goal of establishing a national reference center for citizen science. If we succeed in doing this, we will leave another important legacy, which is a place, which is a node in the network of citizen science projects across Europe.
Thanks, Francesco. I totally agree with you that, of course, public engagement is a crucial part of our job as NBFC, but not only of our center, but all other national centers and universities too. In my personal opinion, also the right word to use is not to convince people of something, but to engage people in a dialogue, so in a productive dialogue, foster public participation in science. So I think citizen science is very important in doing that, if it is done in the right way, of course. And so I also think that the Biodiversity Science Gateway that we have built is also very important to reach this goal, so to foster public participation in science. So can you tell us more about this legacy probably our most tangible legacy as NBFC.
Yes, if building on human capital and engaging the citizens might be considered a sort of intangible product, legacy of NBFC, probably the Biodiversity Scientific Gateway is the most tangible product of biodiversity. The Biodiversity Scientific Gateway, which has been planned 2.5 years ago at the beginning of the project, is now a reality. It has been published. It has a place in the digital environment. The link is down on the right bottom of the slide. And the Biodiversity Scientific Gateway has been thought as a place where researchers, which is fed by researchers with the results of their activity, and is a place where the rest of the world can enter, is something by which the researchers offer the knowledge they have produced to the rest of the world, to the community, local, national, international community. So all the material of different sorts, knowledge, digital applications, checklists, anything which has been produced within the framework of the National Biodiversity Future Center may become something which, a piece of data which is stored in the Biodiversity Scientific Gateway. And this includes data, databases, for example. This includes interviews. This includes stories which are, told by the researchers themselves. And the overall goal of the Biodiversity Scientific Gateway is that of valorize the research in terms of outreach, in terms of communication, in terms of creating new jobs. There is a lot of talking about green jobs, blue jobs, if we look at the sea, and also a way of valorizing the research productions for the businesses, for the private sector. One of the untold goals of the Biodiversity Scientific Gateway is that the private sectors, the businesses, can find within the Biodiversity Scientific Gateway ideas which they can then explore to produce a value out of the knowledge which has been produced by the researcher of the National Biodiversity Future Center.
Thank you, Francesco. In the Biodiversity Scientific Gateway, you will find many things that will be also helpful for your future research, as many of you will become postdocs in the next future or to continue your PhD thesis. And because also in the gateway there will be also all the data that we collected, right? And I guess the next slide is about the digital platform. So can you also tell us about those things that will be implemented within the gateway?
Yeah, again, this is something that we have been working on since the very beginning. We identified these 4 platforms, then We changed the way we refer to the platforms by referring to a unique platform divided into 4 pillars, but this makes very little difference. And again, these platforms now are reality. They will be linked to the scientific gateway. And at the very beginning, we focused on 4 different issues. The digitalization of the collections, which are stored in the national history museums across Italy. We know that Italy doesn't have a national natural history museum, like London, Paris, even New York. But we do have many small natural history museums which are into the universities, the academies, small cities, sometimes within the municipalities. And these small or larger museums are treasures in terms of samples. So we thought that a very good contribution of the National Biodiversity Future Center could have been digitizing, for example, all the herbaria or many herbaria across Italy in order to provide a huge database of these samples. And these samples not only give us a hint of the current biodiversity, current plant biodiversity, but even of the ancient biodiversity, because in some cases, these samples are 1 century or 2 centuries old. Another pillar of this platform is molecular biodiversity. We all know now that studying biodiversity must also include, the study of biodiversity must also include the study of genetic diversity within and among species. A third pillar is dedicated to the exploitation of molecules found within mostly plants, but not only plants, trying to transform them into products, drugs, which can be used in pharmaceuticals, for example. And finally, we will be compiling a list of ecosystem functions which are necessary and which can be an example of how diverse biodiversity is in Italy in terms of ecosystems, and how diverse are the functions that these different ecosystems actually produce.
Thank you very much. And also, maybe it's the last one. So also last week in Milan, for those of you who were there, much have been said about the key exploitable results. So can you mention something about nature-based solutions and all this aspect for the legacy of NBFC?
Sure. We should all remember, this is something that probably many of you do not know, that the initiative within which the money for the National Biodiversity Future Center was dedicated, was taken from, was called at the beginning of the Next Generation EU, the PNRR, the famous PNRR, was called From Research to Business. So in some way, the ministry want to invest the money to create some sort of value. So one of the ways by which we can try to produce value, even economic value, from our research is to define key exploitable resource, KERTs. Now, this is one single example which is quite well developed. It's at a reasonable stage of development within our initiative, which is the directory of nature-based solution. You all know what nature-based solutions are. They are solutions taken from nature to solve problems. So the network of the researchers of the National Biodiversity Future Center worked on a repository of different nature-based solutions. And this repository can be searched by an external client, which is, for example, looking for a way of solving the problem of carbon sequestration, or solving the problem of planting a new forest, or reforest, or solving the problem of the reforestation of a given urban area. So whatever the problem one has in front of you, of him, a solution can be found in this repository of tools which can be used to solve one problem using solutions based on nature. And this will hopefully give a great impulse in supporting further research in biodiversity, because it demonstrates that some of the solutions to the problems we have are intrinsic with nature. And if we share the knowledge of tools that has been already developed, then we can manage to solve some of those problems using nature efficiently.
Thank you. I guess we try in this 30 minutes to give you an idea of what we think are the perspectives for the future of the work we have done in these 2.5 years. So I think we can say that all of these are also opportunities for you. And but I think we can also take some questions for if you have any curiosity or whatever to ask, mainly to Francesco, also to me and Chiara.
Thank you. Thank you so much for the overview. It's very useful, I think, for all of us to get to know all these legacies organized like this. I have a question regarding the Biodiversity Scientific Gateway as an instrument, as a tool to reach out from the scientific community, but do you believe it can also be a tool to involve a citizen in science, not just to get to learn the scientific word, but also to create that space to talk about biodiversity and territories for policymakers? I don't know if I explained myself.
Sure. This is tough, but let me tell you, first of all, that the Scientific Gateway will have 2 physical places. So it's not only a digital device, it will have 2 physical places, one in Palermo and one in Venice. Those buildings will be places where citizens can go and learn the research and the results of on biodiversity. But indeed, the platform with the citizen collecting citizen science projects will be linked, will be part of the gateway. And again, this can be a tool for the policymakers to understand how and when diversity can be, biodiversity of course, can be preserved and restored. But indeed, the most difficult step is to reach policymakers. And when I say that we have a chance of convincing the policymakers about the importance of preserving biodiversity, if we convince the citizens, we know that this is not automatic. But even if it's not automatic, we believe that it's still better to try going through the citizens. Then we will probably need some channels, some communication route directly with the policymakers, the ministry, for example, through which the scientists can provide their points of view, and the policymakers hopefully will listen to them. But, you know, in order to have a debate, we need that the counterpart is willing to listen to us.
Thank you.
Time? Okay. So let me close with the final claim. Even only this morning, I believe yesterday as well, we have been talking about animals, plants, microorganisms, fungi. So I believe a good claim, final claim, could be this one. Every species counts. All of you who are dealing with the species, you know, all the species are important for biodiversity. Thank you very much. Grazie a voi. So I would like to thank Chiara and Fabio for being partners to me in this part of our program. And we now move to the next session, which I have the privilege of sharing, which will include contributions from Giulia Capotorti, who is here with us, Andrea Pilastro, who's here with us as well, and Elena Gissi. And I ask all of them to sit here next to me. We also have two colleagues who should be online, and these are Simonetta Fraschetti and Donatella Spano. So yeah, I know. So let me make a brief introduction by reminding that I actually said this before, how climate change is probably one of the most important, if not the most important issue, also in terms of biodiversity conservation. There is a lot of discussion about climate change, and of course, climate change negationists are there. But we all know that climate change is reality, and we all know that the speed by which climate change proceeds is something that required to do something. We can do something by trying to stop or slow down the speed of climate change, but we can also do something by studying the effect of climate change on different ecosystems, on different species, on different physiological aspects of species. And the goal of this roundtable is to discuss about these issues. So, good afternoon, Donatella. Good afternoon, Simonetta. Thanks a lot for joining us in this roundtable. You are missing a fantastic location here where the PhD Day is organized in this 2025 edition, but you will see it next time. So we are ready to start, and the first to give us her contribution will be Donatella Spano. Donatella Spano is from the University of Sassari, and she will talk us about biodiversity and climate change in general. So introducing from her perspective the topic. Donatella.
Yes, thank you very much for the introduction, and thank you for inviting me, and I'm very sorry for not being there, but I was a conflict, so I couldn't do nothing better. Let me say, if you see my screen.
Yes, it's perfect and we can hear you very well.
Okay, thank you very much. So, my contribution at least is to set the scene, as Francesco said. So, talking about when we talk about climate change, we talk about a lot of issues, not only climate change. And I'd like to start from one point, that is that the main challenges that we are running now is that the global land squeeze. So humanity is putting unprecedented pressure on Earth. And so we have to consider that this pressure is increasing competition for land and for resources. So what is happening is that the land, we need more land to feed people, for energy, for shelter, 10 billion of people by 2050 under these business as usual scenarios. But at the same time, we know that we need to protect and to restore forests and other natural ecosystems. So the main commitment of scientists and for governments is to find the balance between these big issues. So avoid to continue to squeeze our land. So to do that, we need to develop climate resilient environment. And when I talk about climate resilient, I'm saying that the physical part cannot be separated from the biosphere. And that means that biosphere integrity is a precondition for biodiversity and human and social health. And Francesco mentioned this part, this concept. So when we talk about climate change, we think about mitigation, so avoid to emit more CO2 and other gases in the atmosphere. And we talk about adaptation strategies, so how to produce, how to live using options that can adapt to new climate. But this is not sufficient, and they fail if we consider the perturbation that one sector has on other sectors. So, because we are in front of a PhD students and there will be the future generation of researchers, we completely know and we can state that we'll need to invest in advanced and integrated research. So one of the most important feature that was published 2 years ago was to show the interplay of boundaries, planetary boundaries, in particularly how all the process that influence the entire Earth system plays, and how strong is their influence. And in particular, biodiversity loss and the climate change are the core boundaries, are considered the most important process in the planetary boundaries. So it means that when we talk about climate change, we have to talk about biodiversity. So the climate and biodiversity are interdependent. And this is an important concept because we cannot talk about one crisis without talking about the other crisis. So they are called the twin crises. And they are twin crises because they have the same main drivers, the same direct and indirect drivers, and they impact on people's quality life and on Earth, and they are due to the same causes, that is, the human activities. What's the problem? The problem is that there are no common policies for the twin crises. So there is need for innovators, for policies that consider the two issues together. Another aspect that we need to consider in research and studying climate change and biodiversity is the biodiversity conservation strategy must be formulated together. So the biodiversity conservation strategy should be formulated in the context of climate change, and that the strategy that we use to mitigate climate change should recognize and consider the impact on biodiversity. That is important because we cannot use strategies that can have a negative effect on the others. So addressing the two crises simultaneously required that we consider the interconnection and that we identify synergies and trade-off. So action to halt biodiversity loss generally benefit the climate, but it's not always true the opposite. And so in general, the solution requires joint governance. And this is particularly important because we if we consider our Earth, it's full of different landscape with large intact natural areas, but large nature, a mosaic of nature with the people, and also completely modified ecosystems and parts of the land. So maintain the biodiversity requires conservation and restoration efforts that must be coordinated with adaptation and innovation action at all scales. So it's not, it's important that all scales were considered from very local to global. So we need to consider the two crises in a new paradigm. So where we have an habitable climate, a self-sustaining biodiversity, and good quality of life. So it's think that when we plan, when we program, when we think about conservation, restoration, intervention, we consider all multi-functional scape. So altogether land, fresh water, and marine. And this is in some way what Francesco showed, talking about our National Center, because we cover all the scapes. And so if we think that there are measures that are exclusively aimed to mitigate and adaptation to climate change, and they can have a positive or negative, direct and indirect effect on nature and ecosystem services. And in particular, for terrestrial and marine systems, there are climate change mitigation and adaptation options based on nature, as also Francesco mentioned, and technologies that help limit biodiversity losses. And so there are a list of actions that can be taken to protect, to manage, to restore, to create condition to deal with the twin crisis, and they can be considered together. So reduction emission from deforestation and forest degradation means also to avoid, to halt biodiversity loss, and this is just an example. Urban greening and biodiversity support help also the life in city, for example. And restoration of degraded ecosystem has the same positive impact on both climate change and biodiversity. We need to integrate climate, biodiversity, and good quality of life. And that is the solution. And so what we have to ask with a lot of effort is to be to create together the best situation to get the co-benefit from both options. So that means that we need parts of the solution. And from all segment, category of the society, we can get positive actions. And if we think about the multi-actor and multi-scale governance, we are talking about the most appropriate approach that can manage the multifunctional landscape at a different scale. So what we are considering is to help to bring transformative change for sustainable and resilient future for nature and people. And if you consider that the main global challenges, because it's global, the climate change mitigation, because we need to avoid emission, and that can be only global, but for the other main global challenge is to halt biodiversity loss, and this can be only done if we reverse in the curve to maintain ecosystem services, and only if all the options we can get in place can be accompanied by sustainable modes of production and consumption. So a different lifestyle, a different mode of production that can be summarized at bioeconomy. So I think that the main message is this one, and when we don't have to talk about only climate change, that is one of the most important crises, as Francesco said, but it's absolutely deal with the other crisis that is biodiversity loss. I think I can stop here for now.
Oh, thank you so much, Donatella. I forgot to, in my introduction, I forgot to tell you that Donatella Spano is one of the scientific coordinators of Spoke 4 of the National Biodiversity Future Center. She's dealing with, again, terrestrial biodiversity. Donatella, can you say something more about the sort of dichotomy between mitigation and adaptation? Should we rather spend efforts and resources on arresting climate changes or on adapting ourselves and stimulating ecosystems to adapt themselves to climate change?
So the main problem is that the mitigation depends on the politics that every country is adopting, and especially in this period is not a good example of subtracting, of absorbing gases from the atmosphere. The last data we saw for 2004, 2024, sorry, was that it was the peak of the emission, the gas emission. And this is not in particular due to the increase of emission, but the fact that the atmosphere sink is not able to contain gas as the same efficient as in the past. So the problem is that we need to maintain atmosphere, land and ocean efficient to absorb the gas, the climate gas. So we need to climate change gas. So this is the main problem. For adaptation, we have different solutions, and but it means that each sectors, like productive sectors, like agriculture, industry, etc., has to adapt and has to change the way they produce. And this is a big problem because they need to be supported by funding. And they needed to support it by also new guide because the enterprises has to know how to do. So they need science, they need scientific-based data, and they need also some funding to reach it.
Thank you very much. Thank you very much, Donatella. Stay with us, and if you want to intervene again, please raise your hand or make some noise. So we move on to Simonetta Fraschetti. Simonetta Fraschetti is a marine ecologist from the University of Naples, and she will give us her point of view on the relationship between climate change and marine protected areas. Simonetta, thank you very much for being with us.
Thank you very much for the invitation, Francesco and all, and I'm very sorry not to be there with you, but unfortunately, well, there are too many things going on, basically. I do not have a presentation, and you are lucky, because in this way I will be very short and ready for possible questions. And yes, I would like to talk for a few minutes about the potential importance of marine protected areas in this role of limiting the effect of climate change. So starting from the definition, what are marine protected areas? There are so many different definitions around, but let's say that they have the primary objective of the conservation of nature, and are basically a primary area-based tool for marine biodiversity conservation. So conservation of biodiversity, and so with the idea of increasing species richness and the number of different variables is really on top of their objective. There are many other forms for, let's say, protecting or managing the marine environment, but and maybe Elena Gissi will refer to them, from locally managed marine areas to fishery management areas. But let's have a look to those areas in which biodiversity is really the first topic. Another comment before going through, let's say, straight to the topic, is that in the European Union, when the Commission refers to marine protected areas, basically they think to Natura 2000 sites. Natura 2000 sites is it is really the biggest network of marine protected areas, but while in the terrestrial environment, the conservation of species and habitat of European importance is, I would say, in a good shape for many of them. In the marine environment, and especially as far as Italy, Natura 2000 sites are basically without any management body. And so now I'm going to talk about nationally designated MPAs, those that in Italy, but also in other countries, have a well-defined management body. Okay, so these are, let's say, the baseline. Then we have a lot of information from marine protected areas, because a lot of the scientific data comes from the study of these very special places, since we can say that they are really natural laboratories, open laboratories in which we can test a lot of hypotheses about the effect of gradient of human uses, because when they are well managed in the same place, in the same areas, we have areas in which protection is full, and then a gradient of human uses that are more or less regulated. So marine protected areas in the last 30 years have been collected a lot of information. The scientific communities have worked very much on them, and also considering different socio-ecological contexts. So let's say very often the same experiment, into brackets, replicated along latitudinal gradients, but also, let's say, not only environmental gradients, but also socioeconomic, different socioeconomic contexts. Okay, so the result are many and promising. The point is that when a marine protected area is effective, well, there are no doubt that you can really see very important results compared to unprotected areas, because you have always to conduct these experiments in a comparative framework, and so you can see that the diversity increase, the abundance of the species increase, the individual body size increase, the reproductive output. I mean, very important result, especially on some ecological compartment like fish, more than the seascape for a number of reasons, again, compared to unprotected areas. So the point is that when they work, the results are evident, and they are evident not only from, let's say, an ecological point of view, but also from an economic point of view. Porto Cesario, I always refer to these big protected areas in Italy. There was an estimation, an assessment of the economy that was basically driven by the presence of the protected areas, and they were estimating something like 72 million euros coming from sustainable activity every year. In Torre Guaceto, which is a little bit smaller, but very, very effective in terms of ecological response of the protected ecosystem, for €1 invested in conservation, the return is about €3. So, I mean, impressive result. But now I was just referring to the structural variables, okay? Now, the presence of marine protected areas can stimulate also very important processes like stability. And here we have a lot of students, very good students in ecology, and I'm sure that you know that a lot of studies have been carried out to see the effect of biodiversity on ecosystem functioning. So, for instance, on processes like resistance and resilience, and also in this framework, and maybe Boris Worm in 2006 on Science was one of the first showing that marine protected areas can provide more resistance, more resilience, and less temporal variability within protected areas than let's say, the outside framework. And so temporal variability decreases despite the presence of pressures, including climate change. Lisandro Benedetti Cecchi very recently, now I cannot remember that has been published this year or the last year, but they provide evidence that networks of protected areas can have very persistent reef fish communities in very warm oceans. So in those areas in which thermal anomalies were also observed, the fish community persisted. So So if the question was marine protected areas can be an efficient tool for increasing biodiversity, yes, increasing biodiversity translate into, let's say, promoting stability, so more diverse communities can be less affected by climate change. But if the question is, we can solve the problem with marine protected areas? Absolutely not. I mean, they are small spot. They can be considered an important network, sentinels for monitoring changes in space and time of biodiversity, and this role should be better framed for sure. A couple of last messages and then I'm finished. It is very important now what we are going to do in reaching the target 30 by 30 with the 10% of strict protection. Italy has something like the 15% of territorial waters under protection regime. We have to reach these 30%. We have to do it in the proper way, using the appropriate knowledge, and for sure the National Biodiversity Future Center did a lot to improve this knowledge. And this idea of including climate refuge could be for sure very important in terms of implementing protected areas where they are less exposed to thermal anomalies. But in general, I would say as a final comment that we have to change our vision, our politics, because if not, well, as you, Francesco, and Donatella were saying, it will be very difficult to change this trajectory of loss of biodiversity using this very important tool, but they are not enough. Thank you very much.
Thank you, Simonetta.
I hope that I didn't go out of time, I hope.
No, that's okay, that's okay. One quick question. From your point of view, do you think that the marine environment is less resilient or more resilient than the terrestrial environment to global change, to climate change?
At least on paper, it should be much more resilient because it is in the position for a number of structural and functional variable to buffer the changes that are very fastly occurring in the terrestrial environment. The point is that we are going too fast. We are going too fast, and we are going over the threshold, those thresholds that can be, let's say, a safe space even for the marine environment. So my impression, for instance, without going too far, since I'm going into the sea since, I would say, unfortunately, 30 years, is that the 30 first meters of our coastal environment is gone, which is not a good message from my side. I mean, there is a decrease in biodiversity from 0 to 30-meter depth, which is incredible. We are not, last year we went to find some species because we are experimental ecologists, and we were there to manipulate some species. We didn't find the opportunity to manipulate anything because these species are not there anymore. And the disappearance is at the regional scale. So the point is that, at least on paper, and considering the features of the environment, yes, it should have been more resilient to a number of pressure, but we are going too fast. We are going over the threshold. And so we are really in peril.
And yeah.
Thank you so much.
And Francesco, it means that
sure.
the ocean is not doing the work as sync as it should be. That is the other.
Exactly. So the marine environment is supposed to become, to be a buffer. I think we can use many different terms, but the point is that now, well, we are going really too far, but acidification is another big issue, and acidification is something that marine protected areas cannot prevent.
Okay, so we stay on the marine environment and we move to Elena Gissi, who is a, right? who is a marine biologist at the National Research Council and in particular at the ISMAR Institute. Elena. Whatever, or with this.
I change the slide.
Here.
Okay. Okay. Okay, thank you. Thank you for the invitation. So I'm going to propose you some food for thoughts with respect to what we are doing in spoke number 2 of the NBFG and the research I'm doing on climate-smart marine spatial planning. So I'm trying to enlarge the view from individual marine protected areas, but trying to give a perspective with respect to what we can do, how we can act to support biodiversity at a larger scale. So the marine environment is under a huge change, as mentioned by a previous panelist, and the situation that we are dealing with is this one, in which climate change is affecting species, habitats, and so on, but as well human uses on the entire marine environment. And in this context, we need to act to reverse biodiversity loss while trying to adapt and to mitigate climate change. So in this context, marine spatial planning is the process to try to allocate uses in the future. So is an sectorial planning process in trying to allocate uses in space in the future, so trying to achieve economic, social objectives while counteracting and limiting the negative effects of the different maritime activities. So in this context, what MSP can do is this loop potentially. So there is the anthropogenic climate change that is affecting ocean conditions and ecosystem structure and functioning, on which we derive marine ecosystem services for different human uses and activities, but the same human uses and activities affect ecosystem structure and functioning by local anthropogenic stressors. So marine spatial planning tried to act on this framework and to trying to mitigate the combined effects of climate change and local anthropogenic stressors, because we can try to adapt and reduce the stress that comes from local anthropogenic human uses. On the other hand, maritime spatial planning can allocate space for new marine protected areas, so trying to protect areas that maybe are going to maintain the conditions in the future. Also, it can try to mitigate the effects of climate change and reduce the CO2 emissions by producing energy from renewable sources. So trying to include these uses while allocating uses at sea. So, but I would like to stress exactly one point. So within the task number, so we spoke 2 in task number 4, so the project is called MSP for Biodiversity. We tried to mainstream biodiversity in MSP. So how we address especially the point about adapting existing and new marine protected areas within this framework. So we focus on climate refugia. So climate refugia are intended very generally as areas that persist in providing ecosystem services and persist in the present conditions. So we probably buffer climate-induced changes. So what we can do is to try to reduce cumulative effects from multiple uses in these areas that are going to be essential for biodiversity.
But how do we define? What is the definition of this climate refugia? So we know the species will change in their distributions, in their ranges, and they are going to adapt. So there are plenty of definition of what climate refugia is. And the first point of discussion for you is, under these many definitions, I would argue that all these different definitions can give us insights about how to address biodiversity within MSP. We can define climate refugia from the point of view of disturbance, so defining refugia as locations that are less disturbed but different climatic stressors. Or we can take a biological definition, so from refugia defined at species level and in situ refugia, where species will persist, or ex situ refugia as locations that become available for species that are going to change, are going to adapt, are going to occupy different niches. Or we can take another level of definition of climate refugia, so we can try to see where multiple species or multiple group of species will persist in space and time in the future, under future conditions. Or what we are trying to do also in research, also to try to define refugia in terms of areas that are going to provide and maintain ecosystem functioning, to provide multiple ecosystem services independently of the species. I would argue that all these perspectives are needed if you really want to try to understand how to act, how to support biodiversity. So what we did in what I'm going to show is the first part. So I'm going to show how we modeled disturbance refugia, so using a metric which is climate velocity. So we're trying to understand what will be the exposure of marine ecosystems to climate change, so to try to adapt human uses in the future. This is my last slide. So in task number 4, what we did with the MSP for biodiversity, we modeled on the left climate velocity, which is a metric to identify on the water column areas that are gonna change faster than others. So the reddish points are the ones that are gonna change faster in the next 30 years, while the purple dots that you see are areas with no analogs. So areas where there will be no other areas with similar conditions in the future. So potentially we are going to lose these systems. And we try to do this in 3D, trying to understand not only the epipelagic, meaning the main 200 meters of depth, but also to try to understand different on the water column, how the environment, how the ecosystems will change. And then what we did is that we compared, so we tried to understand, we assessed the different distribution and different zoning of marine protected areas and other area-based conservation measures that were defined with a participatory process. And in fact, with MSP for biodiversity, we have been working with stakeholders and with many scientists in trying to design future scenarios of maritime uses. So what we are trying to do now is to assess the different zoning of MPAs with respect to exposure to climate change. And this is done for 2050. So the scheme that you see on the right is a projection of the design of marine protected areas and other ACMs in 2050. But this kind of approach can also help us to assess other activities that depend on the proper ecosystem functioning, for instance, fisheries and aquaculture, but also to try to control maritime transport and energy, so the maritime uses that might affect the ecosystems, especially in the areas that are going to move, that are going to change slower than other areas. So I just want to conclude this here. So with the main messages, we are trying to, with other scientists, we are trying to assess the future scenarios of maritime uses to try to guide decision makers. And just to complete the answer of a previous questions, we just spoke to the task 4. There is an activity which is we tested a transdisciplinary center for marine spatial planning. So which should be as exactly a center to support decision makers in trying to get to understand the knowledge that we are producing within the various spoke, and try to condense this knowledge in messages and then try to build with the decision makers the knowledge that is necessary to take actions from now on. Thank you.
Thank you very much, Elena. In your experience, when species move to multi-species refugia, do new relation between species arise? For example, I don't know, prey, predators, or any other kind of ecological relationships between species?
Well, this is a vast field of investigation for our young researchers. And yes, the answer is yes. And that's why the best way of assessing, for instance, climate refugia is trying to not only look at individual species, but try to understand how prey-predator relationship will adapt or will change. What we saw in this study that I haven't presented here, this under preparation, is exactly that what we see is a change in functional diversity. And so we expect that there will be a change in the way that ecosystems functions. And the best is exactly not to only look at species individually, but trying to work together and to try to see how this relationship will change as ecosystems are going to reorganize themselves under these changing conditions. So absolutely, yes, there is evidence from research, and this is a challenge, methodological challenge also for people studying ecosystem functioning, because it is not that easy to from the methodological point of view, to develop in the field studies trying to understand and to get this relationship. But yes, what we saw already in the Levantian Sea is that ecosystem function is changing, and there is also some substitution of species in specific roles within the trophic cascade. And so this is something that we need to understand if you want then to support ecosystem services and to support the people depending on these systems.
Thank you. Thank you very much. So now we move to Giulia Capotorti. She's from the La Sapienza University in Rome. And maybe she will say something about adaptation to climate change.
We will focus on adaptation now. And thanks to Professor Saspano that already said that biodiversity and climate strictly interact across scales. But due to this cross-scale interaction, we have to consider hierarchical relationships between scales and also cross-scale interactions. So because climate and climate change are not linearly correlated with biodiversity, we have to be aware of the limits of upscaling and downscaling processes. It means that we cannot take the global frameworks in order to put actions at the local level and vice versa. So we have to take into account the overall environmental heterogeneity, not just that provided by climate, but also that joined to lithomorphology or hydrography. We have to be aware that some phenomena occurs just at single scales, while emergent properties arise for ecosystems with respect to single species, as already said before. So very rapid overview of what we know at the different scales. These are examples of current situations, scenarios, and expected responses at the global and regional scale. And the pros that we have available, a lot of data, a lot of information that is also free. So we dispose of quite robust, broad context frameworks, and with acclaimed also fine grain, fine resolutions of data. But in the reality, these data are mainly inferred, not observed, so we have to take into account this limit of this data. We have varied confidence of projections, and these confidences are always declared, so be aware of what is really known and what has to be confirmed. And also, there is a lot of uncertainty about thresholds, and thresholds are important. We already said it this morning. What is uncertainty about, especially about the complex systems? So we know very well some effects for some species or some group of species, like, as in the example in the left down corner. But also we have to consider combinations with the other threats, with land use change, for example. And this is an example on how downscaling may provide not so robust information in our country. And these are two bioclimatic maps provided by the same research group from the University of Ancona. In the left one, we have a downscaling of the WorldClim database for determining the bioclimate in Italy. On the second map, instead, we have mapping of the same types, but with different basic data that are really observed. As you can see, we have a gap in downscaling the global data, especially in mountain areas and in the Mediterranean sector. And this is another bioclimatic map provided by another working group, but in this case, the differences are not due to a downscaling exercise, but to different approaches to spatialization of data. The left one is based on spatialization of indexes, and the second instead is based on spatialization of raw data. And also with a special emphasis on not just latitude, but also combination of latitude with altitudinal gradient aspect and distance from the coast. So we have to integrate different environmental conditions to have a clear picture at these scales. And whatever the result, what is interesting in our country is the transitional zone. We have very biodiverse, we have a huge diversity also in the environmental factors. So in our country, transition is a standard. And what is a transitional climate is a condition for which we have a little summer drought, but we have especially spring precipitations that allow ecosystem to be more resistant during the summer drought period. And the asterisks put in relevance where this transitional environment mainly occur. At the local scale, again, we cannot downscale national data at the local level. This is an exercise that we made for the climate adaptation plan for the municipality of Rome. The landscape represented is the provincial territory, the metropolitan city of Rome. And what we observed with real data, with monitored data, is an increasing trend of impact of climate change on biodiversity sensitive thresholds, moving from the coast toward the inland, according to an altitudinal gradient, the distance from the coast, and also we have to take in consideration also the urban heat island effects in such an environment. So, in this transitional and projected transition for climate, we have to also consider what nature already put in place, that is intraspecific level ecotype diversity. I think the next speaker will talk about this problem, this issue. And I already showed you this morning this slide. We have a single species but different populations that are well adapted to different climatic conditions. And we have also adaptation at the species level. This is an example very interesting that we have semi-deciduous woody plants in our flora. So plants that are well adapted to resist to summer drought, but are also well adapted to winter frost. And at the ecosystem level, we have interesting example of mixture between deciduous and evergreen species. So these are the solutions that nature already provided to transitional environment that can be also adopted for projected transition in climate conditions. And so space for research again for you. This first note is just a suggestion. Try to retrieve, integrate, and update data on current climate conditions and detected or projected changes at the right scale or right scales according to the objective and the targets of your research, by also considering other important environmental factors, such as the availability of water on the ground that can compensate for aridity from the atmosphere. And as regards risk assessment, we have to improve knowledge on critical thresholds, why and where and at which level of increasing temperature ecosystem can experience risk to collapse. And this at multiple levels, so at the intra and interspecific, but also and especially at the ecosystemic level. And also look for the synergies among different threats. Climate change usually combined with land use change, invasion, and the other threats. As regards adaptive responses, and here we come within the framework of the National Biodiversity City Center, we are already testing and monitoring different models for restoration measures that take also into account the projected climate change. So in the right side, you can see a model for forestation that trying to test different patterns and different combination between species in forestation interventions. But we can also have to deal with urgent responses that have no time to wait for results from monitoring and research programs. So in these cases, we can move from general nature-based rules that should ensure resilience, in this case in terrestrial environment. And these rules include give enough space, nature needs space, also adaptation needs space. Try always to think about species diversity. Do not plant a single species for recovering a forest. And these species have to be choice according to biogeographic consistencies and also ecological consistency with local conditions. And also taking into account the importance of ecological connectivity, as we said this morning, because we have to facilitate movement and migration of species by themselves. Thank you.
Oh, thank you very much, Giulia, for this insight into adaptation. So are forests easily adapting to climate change, or they are reluctant to adapt themselves?
They probably will adapt by themselves. We can assist this process, monitor, and try to understand if we are passing the thresholds, and then we have to act in a more active way.
Thank you very much. So our last but not least panelist is Andrea Pilastri. Donatella.
Yes, if I can
sure.
add one thing. First, a suggestion about the use of downscaled data. I usually prefer to use physical downscaling than statistical downscaling, and there are some products that are quite detailed using physical downscaled data with biocorrection. So this is one point. And the other one about your question, I agree. What I can say that mixed species forests in general are more suitable for adaptation than single species, as was also showed in one or two slides before. I think that Julia can agree.
For sure. And I think this is a good frontiers for research now to understand in depth what is occurring in each site, and also to have different solutions that can be inspired by nature. So nature-based solutions have to take into account also these kind of adaptations.
Yeah, we are working on permanent experiments on that. So with the combination of different species, from monoculture to different species together.
Yes, we will communicate by the gateway, I guess.
Yeah.
Okay, so Andrea Pilastro from the University of Padua. He's a zoologist, and he's going to give us some hints on the effects of climate change of one physiological trait, which is reproduction. We see the big face of Charles Darwin. Maybe we can recall that here in Padua, Giovanni Canestrini was the first one to translate The Origin of Species into Italian back 150 years ago.
Okay, so hello everybody. What I'm trying to do very briefly, so don't worry, is to suggest a different perspective from the main approach that conservation biologists usually have when they, work with biodiversity conservation, which is typically an ecological approach. So ecology is a way to see how living organisms interact, making an assumption, which is that they don't change in time, so they don't evolve, which is a reasonable assumption because most of the cases, the processes, the phenomena, the patterns we look at are on a short time scale over which we can safely assume that organisms don't change, don't evolve. So then my point is that in the case of climate change, incorporating a bit of evolution might be important. So I try to convince you and hope some of the young people here might look at what it's doing with a different eye. So we know that biodiversity is threatened by several different anthropogenic factors, like habitat degradation, fragmentation, alien species, and so on. And one of the main threats now is also climate warming, right? Now, there's a huge difference, in my opinion, my view, between climate warming and the other factors, which is that in the short, medium term, we might hope, we do hope, we can reverse or attenuate or even eliminate some of the factors that threaten biodiversity, like exploitation. We can protect species, we can protect environment, we can increase connection between fragmented environments, and so on. But for sure, we know that we are not going to reverse climate warming for a century or more, which means for many species, a number of generations that is enough to expect that evolutionary changes may occur. Then there's another reason. Well, Julia, well, I was saying climate warming is affecting every individual in a population everywhere on the planet. So Julia just explained that's not perfectly so, but it's an approximation as compared to other factors. Climate warming is occurring at a very, say, universal scale. Terrestrial environment, freshwater, marine environment, and for many organisms, there's no way to escape this. So there might be thermal refugia for many animals and plants, but not for all of them. And this means that all organisms in populations out there will have to face new thermal conditions. Now, climate warming has two aspects. One is the change in the average temperatures. Okay, it doesn't work. But anyway, you can see there. So an increase, a progressive small increase in average temperatures over the year or breeding season or whatever. But we know that is inevitably associated also with the increase in frequency and intensity of extreme events that we call heat waves. But of course, there can be other climatic extreme events like rainfalls or droughts, but I'll focus on temperature for the moment. Now, these two facets of climate warming actually have two different selective pressures on organisms because average increase is likely to affect mainly phenotype in terms of phenology, so timing, and spatial distribution, moving, escaping hottest areas. Whereas heat waves are actually challenging, at least in some environments, the thermal capabilities to cope with extreme temperatures, which might affect other types, other genes, genes that are specifically associated with resistance to thermal stress, for example, but most likely also overall conditions of individuals, which might mean, for example, mutational load of individuals, which is related with, for example, the level of inbreeding or the demographic history of populations. I've been listening yesterday, people presenting super interesting work on, for example, using morphology, functional characteristics of plants, for example, in studying ecosystem or populations, compositions, and biodiversity, richness, and this type of aspects. And I think in the case of climate warming, we need also to incorporate perhaps more evolutionary traits like effective population size, plasticity, but also reproductive strategies like R strategies versus key strategies, because we know that in particular extreme temperatures affect particularly reproduction, which means gametes, but also offspring, and the potential of a species to adapt is also determined, in particular when we talk about heat waves, is particularly affecting reproduction, which is good and bad, it depends. But for an R species might be good because then you might have selection at a low cost in terms of demography. And so this is what I was mentioning before. So I think that we might gain further insights in how communities and biodiversity in general might respond to this challenge by incorporating in our analysis not only ecological traits, but also life history traits and the perception or the awareness that climate warming might select different components of heritable phenotypic variation within species. And therefore, my suggestion would be to incorporate life history traits in biodiversity studies, as many of you are doing and conducting so successfully and brilliantly, and also to investigate fitness functions in relation to temperature, both to average temperatures, but also to heat waves. I've listened Filippo, I think, presenting a super interesting study on lagoon plants, zoologists, so I can ignore plants, which is super. And that type of study, for example, might also give hints into the potential of populations to evolve and respond to. And also the using, of course, since evolution is based on heritability of fitness, we might successfully use all those omic tools that allow to dissect and understand what's going on. And this is not just interesting for science, but I think is very important in the case of specific of climate warming to predict population resilience in the midterm, so not in the next 5 years perhaps, but certainly in the next few decades, or even less so. And also can provide important practical guidance to, for example, urban green management that often are, like ignoring processes that are occurring all the time in populations, like selection, and of course inform in situ and ex situ conservation plans. We heard about germ plants, germ banks for plants that are very important, and those are crucial because, of course, you have a small, tiny sample of a population, of the genetic variability of that population, and it's important to be aware of the consequences that this might have, and so on. But this applies also to animals. I think that was the last one, so I just throw a stone in the lake and see what happens.
Thank you, Andrea. One quick question. Do you, in your experience, do you see any differences in the ability of adaptation in different taxonomic groups of animals? Like you show birds.
I just go back to the first slide, because these are two species that we are going to study in the rock sparrow, the small bird. You don't see it. And the kestrel, which is a slightly larger bird. Now, the rock sparrow population we are studying lives on the Alps, and it has apparently been positively affected by climate warming. So they are increasing reproductive success because it's warmer. That is a sort of Mediterranean mountain species, whereas the lesser kestrel, which breeds in Matera, one of those areas that I saw in red, I think is a bad news. But anyway, they are affected by heat waves in the last 4 years, something that never happened before, as far as we know, and they've badly affected. So this is just an example of how different species can be, in Italy, be positively or negatively affected by the same process, the same phenomenon. And the interesting thing is that climate warming is also affecting evolutionary responses in these bird populations. For example, lesser kestrels, we measure selection for heterozygosity associated with heat wave mortality. So it's bad, but it is selecting more heterozygous individuals, which is good for the population, of course, because it increases potential genetic variability, or it reduces less. They are increasing in size. We don't know how to interpret that, basically. And rock sparrows are changing their mating system. They become more strongly polygynous. So there are sexual selection things going on. And I have to also to mention that we have a fantastic experiment that has been done by other collaborators in Padova on the effect of heat waves on sexual selection in guppies, which is a tropical species, so it's not Italian species, but it is very useful to see or to dissect how a very short time of high temperature can actually affect, in principle, effective population size, reproductive bias, and selection for conditions. So I think that, of course, it's very difficult to do evolutionary studies on many species, as we do when we monitor ecosystems or large geographic areas. But to go to your question that I didn't answer, actually, we need to have more of this information from more diverse taxa. I think we have the potential to do that, because I hear about many colleagues working on the type of experiment as the one I mentioned before, like in plants, and for marine species and aquatic species in general. They are very feasible for also laboratory experiments, in which, adding a bit of ingredients, you can get information about, or a feeling about how they might adapt. By the way, the word adaptation we use in evolution biology is different from the use of adaptation, which is typically, and that is something that we probably need to sort out sooner or later.
Thank you. Mariella, do we have some time for, couple of questions? Is there anyone here? First row.
This is probably a question for the last two interventions that we had. And this is more a topic that I've heard in the forestry groups, and also sometimes when we hear talking about reforestation, we hear talking about assisting migration of species somehow. So like, for example, we want to do a reforestation in the Po Valley, and we're going to choose more like species, like trees that, tree species that are more adapted to like warmer climates, and they are more thermophilous species. And and this is something also, I don't know, probably connected to also, for example, Julia, you showed the potential natural vegetation chart. And also I think it's going to evolve in time if the climate change bring us to have higher temperature. So I would like to know what's your opinion in a more ecological perspective, also maybe evolutionary perspective of this kind of practice of the assisting the migration of species. I don't know if I was...
Who wants to answer?
Very complex question. For example, for ecotypes, it has been demonstrated that putting individuals from populations in from the Mediterranean area into forestation activities toward the north was more effective. So we can use native species always, but different populations, ecotypes to respond well. As regard migration, we have to reconnect our natural residual patches because we don't know how and where species can migrate, and we can assist, we can provide the potential to do that. And we can observe and eventually intervene where species cannot migrate or species cannot adapt to the metrics that they should be crossed. This is a problem for the medium level of the ecosystem types because along coasts and on the top of the mountain ranges, the thresholds are already passed, so we have no so many opportunity to assist migration in these cases. We have no more spaces toward altitude, neither towards the sea, because the sea is rising. Le domande di Chiara sono terribili.
No, I totally agree. So we can predict a little bit what might work better. As you suggested, plants from warmer climates might expect it to be. But of course, we know very little about that. And one practical might be to, as Julia said, to allow nature, in a sense, or natural selection to work on. The cost and benefits, but I think that should be a strategy in general. Like for example, for urban areas, we tend to decide which plant we want, to plant one big individuals that costs a lot, and then after 10 years it dies. And so an alternative strategy may be to be more flexible, to use different plants, to plant, for example, in small groups instead of single individuals, and for some reasons that we might never know, one will grow and the other will be swept away by whatever. So.
One more question? Here.
Thank you. And thank you for your presentations. I have a general and open question. It has bothered me a little, and I just wanted to know what you think about it. There's a school of thought that believes we're in a late ice age, and so global temperatures were hotter than they are now. And although species went extinct, some are coming, some are resurfacing, and so many think the climate change now, it's just a propaganda, and even though biodiversity is being lost, it can resurface because was supposed to be in a late ice age, so it's not as dramatic as it was before. I just want to know what you think about that.
Your question, in a way, made me think about the previous one somehow, because in a way, climate change and a bit challenge us to think about in situ conservation. So what is in situ conservation? And I'm saying so because in situ conservation, long-term in situ conservation is one of the criteria that we have to demonstrate if you want to designate an area as other area-based conservation measures. And now we, like, through the Montreal Convention Biodiversity, the Convention on Biological Diversity, we can achieve the 30 by 30 targets, also including OECMs, so area-based conservation measures. So if I understood your question about what is the meaning of let's say, a species will change, will move, new species will get in, and so it's just a propaganda because nature will do it by itself. I would say in a way, as scientists working in conservation, biodiversity conservation, and so on, I think we are challenged. I don't want to enter into fake news, but I want to focus on what is in situ conservation, when species will adapt, will move, will change. What do we think, especially because we see this restructuring of ecosystems, because species are moving, species are adapting, species are evolving. So from a conservation point of view, we have to question ourselves as scientists working in biology and ecology about what we really think about in situ conservation is now under climate change. And looking at, in some part of my research, when I'm trying to see how ecosystem functional traits will restructure, will recombine under species that will change, but the levels of functional diversity will stay. Sometimes I'm questioned. I try to abstract myself from species as the only paradigm through which I look at biodiversity and try to enlarge this view. So I think this question is very critical and challenge us in our research and in answering to decision makers somehow. Because myself, I'm struggling with the sense of, okay, is this, I know what are the objectives that I have to achieve by law, but I also think from an ecological point of view is that probably we are really even too much our view is very strict on the species perspective and not on a community level. And I think there is a need to look at communities, and I will restructure a bit, moving beyond just looking at persistence of species, because then what was suggested is, what if we change species? What we restore communities with different assemblages that were not the native ones? I think we need now, and we are challenged to rethink about the basic construct on which we look at our systems, and then in which we decide, or which we take decisions about conservation in our actions. Woohoo! They agree. I'm happy.
Can I give you a very selfish answer to your question? And again, I'm quoting somebody who works here at the University of Padua, whom you met yesterday, Telmo Piovani. Nature will probably get away with it, with sudden change. Man, who knows? More questions?
May I add something?
Sure.
Thank you very much.
Simonetta, go ahead.
I think that we are already, so 2 points. We are already moving towards an ecosystem perspective, starting from the Marine Strategy Framework Directive. The Marine Strategy Framework Directive is looking for reaching the good environmental status at ecosystem level across all European countries. So we already move first by the one directive that to me is one of the best that has been produced by the European Union, and this should be the target. The point is that at the same time, what we were saying before, so that at 2030 we have to reach the 30 by 30, the 10% of strictly protected areas, the good environmental status, the 20% of degraded habitats and not species, so species, habitat and ecosystem, because the nature restoration law is referring very often to ecosystem. These targets are really ambitious and are to some extent not considering that we are not in the position to reach them, because we are at 2030 25, in 5 years, it will be very difficult to reach these targets because we do not have enough knowledge. And I'm sorry to repeat one issue. Yes, our environment are adapting to some extent, but we are all observing a decreasing species richness. Francesco Bavestrello for Portofino is demonstrating that in 40 years that area was able to lose basically the 0.5% of hydrozoans, and the same pattern was shown for many other species. It's not only for climate change, it is for multiple stressors. So increasing human uses, industrialization, the blue acceleration, I don't know how to call it. So the issue is, we, as scientists, we are not scared about the different strategy that we can adopt for looking to the adaptation, looking at species level, at habitat level. The point is that we really need a new vision, new tools, not new targets, because targets are most of the time just on paper. We need to think about qualities and not just quantities, but we really need large scale initiative to stop this incredible trajectory of loss that we are all observing. This is something that to me is very important. I don't know if to some extent I was contributing a little bit to answering to the question.
We are cutting the branch on which we sit.
Yes.
More...
Yeah, so Francesco, the conclusion is we don't have time to wait but we need to waste time to discuss about people who deny biodiversity loss and climate change that doesn't exist, but we need to act very soon, and also we have a lot of space for research, for studying, for improve our knowledge. So no space for negationists, but space for research and for improve our knowledge.
So from the place from the very place where Galileo Galilei taught everybody the scientific method, we hope we pave the way to solve some of the problems that we ourselves have created. I think it's time to close, so I would like to ask an applause for our 5 panelists. Donatella, thank you so much. Simonetta, thank you so much. Elena, Giulia, Andrea. And I would also like to ask you a second round of applause for Mariella Rasotto, Fabio De Pascale, Chiara Anzolini, and all the staff from the University of Padua who has made these 3rd PhD days possible. You have done an excellent job. Thank you so much.
Thank you. First the guys.
Thank you so much. We are very proud to host this final event here, where the 2 of us graduate for our PhD. So it's like a good hope for those of you that are concluding your PhD. So thank you, and have a nice career.
So it was a pleasure organizing these 3 PhD days in the last 3 years. Now we don't know what the future will bring to all of us, but we hope for the best for you and for us too.
And join me in thanks again, Mariella, that I'm honored to call colleague.
Thank you. So what I have to say, apparently, just good luck to you and to the young one, such as Chiara, Livia, and the young researchers that are inside the NBFG, but they do not know about their future. So good luck from my point of view. It has been a pleasure to see all of you in those last 3 years. I have seen you growing with your research, very nice result, and especially what I have seen is you grow up as a group. You know each other, you have a chat and so on. So it's true, Francesco, we have a human capital that in some way we were able to aggregate. So I hope to see you soon, next year, I don't know, but anyway, good luck to everybody.